And I think I'll be able
I'll be
to.
able to.
There is a
There is
echo.
a echo.
Yeah,
Sir,
the,
not
Let
now.
me
Yes, sir.
reduce.
Please continue, sir. Not
yeah,
now.
please.
Yeah, I'll reduce the. You are able to hear me without echo now?
Yes, sir.
Yes.
We are able to hear without echo. You can continue, sir. Thank you.
Good afternoon, everybody. Uh, I hope I'll be able to awake you from the siesta and your long sojourn to the birthplace of God Krishna, Lord Krishna, on this auspicious Hanuman Jayanti. I'll be speaking on genomic assisted breeding for chickpea improvement. Uh, First of all, I thank Professor Arun and his whole of his team to have given me this opportunity, and thanks to organizers, Indian Society of Genetics, Biotechnology Research and Development, Agra, especially to the staff of Hindustan College of Science and Technology, Farah, Agra. Well, now there is a big challenge before each one of us. With you, the young budding scientists, that our water footprint, which we generally go unnoticed when we start producing food stuff, be it fruits, be it vegetables, be it the dairy stuff, be it the non-vegetarian stuff. Now the water footprints cost us a lot. How do we know also when we go to 2050, feed 10 billion people with less resources, less arable land, less water, less labor? That is the question. And that's the great farm challenge. The question is not that where this produce is coming from. The question is how sustainable is this food production system? Whenever we are speaking of sustainability, we are speaking of conserving our resources, reutilizing our resources, recycling the resources, using lesser inorganic material, and at the same time increasing the yield, because you have almost 10 billion mouths to feed in another 30 years. Not a way much ahead, but going to come in a jiffy. So it is at this place that the pulses, which are the pulse of the country, the pulse of the soil, the pulse of the population. Why I say? Because for a large vegan population, it is the poor man's meat. They provide the excellent nutrients in a well balanced system, and mostly so in case of chickpea, which is said to be the next best complete food after egg. Now, when you look at the carbon footprints, which I have just passed on in the first slide, you understand that chickpea also has very low carbon footprints for the amount of nutrients it produces. It's almost 1/6 of rice itself. Leave aside, it is 1/18 of meat and 1/12 of egg. That means it is going to produce much more nutrients with lesser water and lesser use of the natural resources. Well, it has very nutritional benefits, but when we look at genomic assisted breeding, which I'm going to link up with chickpea, you need to understand that the genomic assisted breeding practices predict better genotypes based on the phenotypic data, or a vice versa, better genotypes from the available phenotypic data. We have genetic resources, and the whole idea is to create improved cultivars, maybe breeding lines, maybe advanced cultivars, and then you have a plethora of information here. Earlier, all our statistical analyses that have been taught us used to directly predict a better phenotype based on the phenotypic variability. Slowly, as the marker repertoire and the marker discerning ability increased, we started looking at the QTLs per se, the marker trait associations, which we called as MTAs, started developing genetic and the physical mapping. A large number of sequencing repertoires have been developed. We also started looking at the transcriptomics, proteomics, and the tilling approaches. The whole idea is to better predict the phenotype from the genotypic information, or better predict the genotype or the genes by the phenotypic information. But if you just don't utilize it, it becomes academia, and that is not the purpose of modern day science. The whole modern day science tries to utilize this genomic and phenotypic information for developing better cultivars. Well, what does a translational genomics, particularly in most of the crops and more so with relation to today's topic, do? It tries to fully utilize the modern molecular genomic analysis methods for agricultural breeding and research programs, and there are certain prerequisites that we need to understand. You need to have a robust breeding program, nothing can circumvent it. You need to have a large scale field screening, well developed, maybe mapping populations, maybe association populations, or maybe targeted trait lines. But most important, what we require are the people, the human resources who can use, who can understand, and who can actually implicate them into the precision breeding programs. You need to have a strong biotechnology approach with an healthy interdisciplinary approaches. Well, this calls for productive collaborations, maybe nationally, maybe internationally, and good funded projects. Now, as my experience goes, where actually I'm speaking of all from my lab, we started analyzing the global chickpea accessions because we found that the base or the genetic base of the chickpea was very narrow and we need to broaden it. We started looking at almost all the plethora of the lines that were available in the Indian subcontinent used by the NARS, National Agricultural Research System partners, and those from the ICRISAT gene bank, which were of the Indian origin, and compared with my program with I.S.G.B.R.D, which was earlier based in Syria, now moved to Lebanon and Beirut, where we got their lines and the wild species and a particular line developed by IRI, which was having wild species and the wild relatives. And we saw that almost these two particular groups, that is the Mediterranean chickpea and the Indian subcontinent chickpea, were distinct two branches, and then we could achieve large genetic gains and broaden the base when we hybridize this. And this has paved way for us to develop certain mapping populations. And when I joined this program way back in 2007, we had literally no maps. A good amount of work was done by the rice group and the wheat groups, the maize and the soybean groups, which I was initially working for. But when I joined in chickpea, we had few maps from the Huttle as well as the winter groups of Germany and USA as well as dollars, where they had good amount of interspecific maps, but we didn't have intraspecific map. And whenever I started using the markers for my breeding program, I basically understood that most of these markers, which were highly polymorphic in the in the map reported, were skewed towards the wild species and within the cultivated. Even now, some of my SRFs like Chetna, we are finding it difficult to find good amount of polymorphism if these are quite nearly related. So we started, this was somewhere in 2011 when I initiated this development of molecular linkage map. We started using a Desi and Kabuli, which are intraspecific group that is within the Aritinum group, and we developed a small map which we had published. And later it was one of the highly cited articles because this was one of the first intraspecific map in India and the second in the globe. And we moved ahead with trying to saturate this genomic regions in this particular map and also develop an high density map and a SNP map almost with 1 lakh raw SNPs were obtained, and we could curate them and make about 3,000 and odd SNPs, which covered about 8 linkage groups, giving about 1k centimorgan distance of 0.33. And not only that, we also identified certain QTL reasons, the marker trait associations, particularly with yield. This was my DBT project along with Dr. Sabhyata from National Institute of Plant Genomic Resources and Dr. Andrew Samand and Raju Dattla from NCBI Canada, Saskatchewan. So we identified a large number of QTLs for various yield traits. This did come out in Nature scientific reports. But then what? This was the time, because I was a basic breeder, I thought that I need to utilize this data into developing bold seeded lines. Particularly, bold seeded are a speciality, particularly in Kabuli chickpea types. Basically, Indians use them for what we call as culinary purposes. Chola, what we start making, and bold seed gets a premium price of almost 1,000 rupees higher when you compare to the medium and the small sized ones. We started evaluating these particular lines at almost 5 locations, particularly into the breeding program, which had good wilt reaction. And we started now developing some extra bold of 66 grams. We call it as medium bold of 56 grams, and the small seed, which says 50 grams. So these are now in the coordinated evaluation, going to see the light of the tunnel at the perhaps by next year. At the same time, one of the dreaded disease in India is wilt, which is caused by Fusarium oxysporum, forma species Siserae. This has various races across the country. Race 1, race 2, we have about more than 8 races, but in India, 6 races have been reported. And this is controlled by generally a single gene, which is a recessive gene. Breeding for it is very difficult. There are pathotypes. And then at no single place, though you have a predominant race, you have other races too. So the varieties which were released earlier deteriorated fast because of this changing race scenario. And to get a durable resistance, you need to pyramid for these alleles. Now, pyramiding for these alleles also calls for evaluating these developed lines at multiple places where multiple races are there, or in microplots which are too small and breeding populations cannot be handled. And most of the lines that have been represented into the all India coordinated trials usually perished due to this fact, and most of the breeding programs suffered. This was when we thought of, along with my friend Madam Teresa Milan in Coloba University, where we could identify the fork alleles, Fusarium oxysporum, as I said, forma species Siserae. We could find that the linkage group 2 had almost 4 of these fork alleles, 1453. That means it covers most of the spans except for fork 2, which is present in central India, and we need to depend on the local varieties. We thought upon improving the chickpea germplasm. We did a post-ante work where we could identify and validate a set of markers, particularly the STMS markers, against 0, 1, 2, 3, 4, 5. 0 is absent in India. So we made a panel of these markers and evaluating our lines. And what we have taken is the WR315, which is the tolerant source. And then we introgress these into a susceptible line like PUSA 372. Only the genomic regions which carried almost all these 4 of the 5 fork alleles, which we could pyramid and present into what we call as a marker assisted breeding program. Now we had a wilt-seq plot at IRI, which is basically for fork 3 and 4. But you can see that the resistant lines, if you look along from the sowing to that of the growing plant, the susceptible lines have died, and we could get a plethora of lines, including single plant selections carrying the putative alleles. We did develop a SNP marker which was showing tag to these alleles, and almost all our lines, when you look at the susceptible lines which did not carry this marker, and the resistant analogs which had this marker along with our lines. No, we didn't stop there. We went to and developing an hydroponics techniques thanks to my students Apurva and Jorben, who developed this rapid protocol hydroponic method where you could clearly identify the susceptible lines from the resistant lines. Not only that, against the subtypes or the pathotypes which are prevalent at each location. So we selected the dominant 2 pathotypes And we could prove through histoanatomical studies that the normal xylem was seen in case of the resistant lines, and in the susceptible lines the fungus has actually colonized the xylem region. And we could now identify novel, for example, ILC Latvia, which is a land race. Similarly, Czechoslovakia varied resistance, and now we are doing the allelic relationships along with WR315. But this also over a period of 8 to 10 years, supported by CIMET, as well as from our DBT program, develop a large amount of lines, some of them in the advanced varietal trial. And when we have done a disease score, it clearly indicated that our tolerant lines, for example, the Latvia had a good tolerance score when compared to the susceptible lines. Now, if you look at this, we could clearly develop and relocate almost all these tolerant alleles into our marker assisted backcross lines. You can very clearly look down that if it is the recipient parent, almost all our MABC lines are highly resistant. Not only that, over 2 years of period against the national checks, this particular line as well as its sister nil line has given very high resistance over almost 8 locations, and then it has been released last year with the name Pusa Chickpea Manav. Now look at the whooping yield advantage that it has been giving. Apart from high amount of protein, almost 19%, but the yield advantage that it was giving was 28% over recurrent parent. More than that, over the national checks, it was giving 36 to 66%. Has been dubbed as a super chickpea variety, both by I.G.B.R.D as well as ICRISAT through my partnership with Dr. Rajiv Varshne and Manish Rurkiwal, who have done the genotyping activities. And when you look down, that it is spreading like a wildfire among the farming community. Now we are mobilizing these QTLs into another mega variety, that is JG11, which has almost 400 quintals of seed production. This is your Pusa chickpea manav. It's a huge strip, absolutely resistant. The donor parent, the susceptible JG62, which almost perished. And this is our new line now into the ACRIP trials 20215. Now, similarly, when we were looking for yeast for an evergreen revolution, particularly in case of chickpea, botrytis gray mold is one of the major bottlenecks of production. Now we have screened again land races because land races actually form a valuable source of genes, alleles, and whatnot. Well, friends, if you look down at the modern large fruit size and higher total soluble content in case of tomato, Lycopersicon esculentum, we didn't get them from our cultivated tomato. We got it from the wild tomato, Lycopersicum peruvianum, which has very small size fruits, but it had the QTLs for large size fruits, greater flesh, that is the greater TSS, total soluble solids, more sweetness. So these land races, which represent in between the wild species and the modern cultivars, actually are a pool of genes that have not been exploited, not only in case of chickpea but in most all the crops, and these need to be relooked, revisited, and therefore they form the precious sources. And now when we are screened both at the cut pig method and also using the bulk sample analysis, one of my student Supriya Sasdev worked, we have already published this in Phytopathologia Mediterranea. We could get some putative MTS, and we started moving ahead with identifying the QTLs in a large mapping population and could identify consensus QTLs, which we started mobilizing into one of our developed line, GL13042. This has been again released this year. It is only the second cultivar in the world that has been developed from what we call as using wild sources as one of its blood, and it has absolute resistance when you're looking at botrytis gray mold. It has also multiple resistance, whether you're looking at Fusarium wilt, it has tolerance, spot borer damage, tolerance, ascochyta blight resistance. So we developed a multiple line which has been now released. As you see, it is a cross between a wild accession Cicer judaicum into Aratinum GPF2, which has been now released as a cultivar for Northwest Plain Jones. Now, one of the another important abiotic stress, we have seen two biotic stresses where genomic assisted breeding plays a major role. When you look at abiotic stresses, drought is one of the major stresses. Different concepts would have been taught even in your classes where tolerance or resistance is governed by several factors. There are various physiological processes or the phenotypic expressions of these physiological processes which are basically governed by the biochemical characters. Some have accredited drought tolerance to root mass index. Someone else has said that it is the threshold of leaf expansion, which is governed by the osmotic potential, again governed by rate of transpiration. Some other workers have indicated the relative water content, membrane stability index, or the canopy temperature depression. Well, everyone understand that there are different processes, genes or QTLs, which govern these traits, and these needs to be pyramided where there is a consensus. Now, since it is like your QTL breeding, the conventional breeding approaches are time consuming. And it's a challenge that had remained unbroken portfolio for the last almost 50 or 60 years of chickpea breeding. We started looking particularly in the central and the northwest plain zone conditions. What are the characters? It is the deep root system, it is not only that, it is the above canopy temperature characters like the membrane stability index as well as relative water content. We developed a mapping population and developed a very well saturated map where the QTLs have been identified. And we basically found that those lines or genotypes which were able to produce greater biomass in the initial basic vegetative phase, that's what this particular indicates. That means if they're able to produce better biomass in the initial basic vegetative phase, like your Pusa 72, Pusa 362, or some of the lines like ICC 4958, then what we look down is the yield differences between the normal or the salt stress, or the yield differences between the normal and the drought stress lines is very less, indicating they have better drought tolerance index. Now we started looking at some of the genes, and we identified the ethylene responsive factor gene, a ubiquitin family gene, uh, and the DREB. It belongs. It also hosts the DREB family group. Almost we could actually find about 120 putative ERF genes which were upregulated, and we could classify them into uh, 11 groups. Now, provisionally or putatively, we could identify the Cicer Aratinum ERF116, which was differentially expressed between the drought tolerant and the susceptible chickpea cultivars through SSH. And then we found that this particular thing actually coded for a putative protein of 241 amino acids, and then it lies at the C terminal region and was playing a major role. We did identify it and characterized it in an heterologous system. And when we did that, we checked out that it not only played a major role in case of drought tolerance, but most of the abiotic tolerance genes. That's why I use the terminology that it's an ubiquitin, whether it is for salinity, whether it is for cold, whether it is for what you call as ripening, and so that is why earnestly it has been reported as ethylene responsive factor. Not only that, the promoter sequence analysis identified presence of several not only abiotic but biotic stress response cis elements. Now we are looking at its biotic stress tolerance and what is its importance. We did publish that it also plays a major role in case of heat. Now, when we look at the two major groups, the Desi or the Kalachana, which you call it, and the Kabuli groups, Desi groups inadvertently had better drought tolerance, particularly expressing this through tertiary branches, the total number of branches and the pods per plant. And we started looking again at land races. Can we identify newer sources? And we could identify some of the newer sources. And we started looking at some of the sequence similarity approach with already well reported genes. We started with almost 120 genes and could drop down to 15 genes. And we looked at different genotypes basically worked differently in different genotypes. For example, in JG14, which is for heat and drought, ICCB 929444, if it is sucrose phosphate synthase, the Medicago pathway, then it can be ethylene responsive binding factor in case of L550 and RSG888, other highly drought tolerant cultivars. Whereas in our ICC 4958, it is again sucrose phosphate synthase Medicago pathway. Of course, the gene ontologies have clearly identified this particular thing. But what we were basically interested is, can we actually identify certain novel sequences and also look at a dehydrin gene, which was playing a major role? And we found that in case of legumes, the DHN gene was highly conserved among the germplasm and played an important role apart from ERF family genes. Then we found a novel gene, ASR, abscisic acid synthesis and ripening gene, which we could allele out from some of the land races as well as our genomic population. And we sequence this. Of course, this is as an NCBA gene tag. And we went ahead with Ramchandran plot structural validation and could find the domain conserving sequence. And when we started looking at the predicted functional partners, it grouped along with the LIA genes, which all of you know play a major role in drought and osmotic tolerance in the developing embryo. Similarly, with the dehydrin gene, that was why we also looked at the dehydrin gene and also some uncharacterized proteins, but all related to water stress under depleted water conditions. So this has led to our publication of identification of an hypothetical novel abscisic acid and stress ripening protein, which was published and highly cited in case of plus one. Now, because as I said, as a breeder, neither in case of wilt, neither in case of drought, we did stop with just publications. We thought that now having so much of plethora of the information and then discerning this particular QTL region and then saturating it with markers, can we now mobilize these QTLs into elite lines? See, we are not speaking of just already susceptible lines where you can get some amount of drought tolerance by mobilizing this QTL. We wanted to mobilize it into mega already drought tolerant varieties and look what is the amount of resistance. And with my partners with Thicrisat under an DBT Australia India Strategic Relation Fund, along with those in the UWA University of Western Australia, Perth, incidentally, where I'm also the associate adjunct professor. We try to introgress this QTL region using a plethora of markers. And this was our donor line, ICC 4958. And the PUSA 362, also we have done it in 372 and 391, and a large number of mega varieties. If you start looking at this cultivar, which is a back cross generation, you can identify that it has the greater root volume and the root length from that of the donor parent and the above ground parameters from the recipient parent. These lines are not just released for academia, but they are tested vigorously by the All India Coordinated Research Program on chickpea. Over 2 years, over almost 8 locations, they also carried the Wilsec QTLs under extreme drought conditions. You can look what is the moisture percentage at flowering. It did not exceed 20%. And at the time of maturity, except leaving the location of Sihor, which had rains over 2 years average, we had almost a 15% if you look at average. That means between 15 to 22%, where almost all the crops flourish. This particular line, BGM 10216, out-yielded all the lines, including the national check, and has been released not by Dr. Bharadwaj, IRI, or IAPR, has been released by Central Variety Release Committee by Department of agriculture and cooperation and farmers welfare for central zone, that is consisting of the states of Madhya Pradesh, Chhattisgarh, western UP, Bundelkhand region, western Rajasthan, Gujarat, and Maharashtra as Pusa Chickpea 10216. Again, thanks to Rajiv Varshne, my close friend who has now relocated to Murdo, and we continue with excellent collaborations. With the release of Pusa 10216, which was in 2020, much ahead of Pusa Chickpea Manav, in 2021. India leads chickpea program in the world, legume program anywhere in the world, and next only to rice, wheat, and maize program of the world. We are the leaders even in case of breeding programs in India to release the world's first MABC varieties, both for drought as well as wilt. And now we are looking at an what we call as convergence program to develop a variety that is tolerant to wilt as well as drought, as well as for Ascochyta blight. And my young researchers are already doing this crossing programs. Not only that, the soundness of this QTL was identified even at Ethiopia, which is a dry area where this particular QTL from ICC 4958 was introgressed into the genetic background of JG11. Now we have a large amount of varieties. Like I said, we have released in Pusa Chickpea 10216. Now another with my colleague Dr. Sailesh Tripathi, Pusa Chickpea 4005 for Northwest Plain Zone. Similarly, IPCL 4-14 again for Northwest Plain Zone. Similarly, in case of wilt, these are all genomic assisted bred varieties. Whether it was Super Anigiri for South Zone or the Pusa Chickpea Manav from me for the Central Zone, or from a friend Dr. Aditya again for wilt. So now genomic assisted breeding, particularly in case of chickpea, has become a routine breeding tool. Now in a span of almost 12 years, from resource poor orphan crops, particularly chickpea, has now become resource rich and the first marker assisted bred crops from India to the world. We were also looking at some of the stresses like salinity stress. My colleague Neeraj and my student who has joined along in this program, we found that this was one of the major problems that is slowly increasing in global. Almost 2% of the area that is increasing, even in India, maybe due to faulty irrigation, substandard irrigation, or over irrigation. Large tracts of Punjab, Haryana, Rajasthan, even certain tracts in Madhya Pradesh where water is in plenty, where faulty irrigation with no proper drainage is going on, salinity is increasing. We started looking. Initially, the seedling establishment is a major problem. Snehapriya and her group tried to establish a rapid screening protocol using the hydroponics, where this technique just not allows you to screen, but also to transplant these tolerant genotypes so that you can go ahead with the crossing program. This also helps you to identify in the segregating generations itself the lines which can further be transplanted and later used in crossing program for a marker assisted recurrent selection, which we should be doing from this year. So we have done the microplot screening. So this was Neeraj's PhD work, both at CSSRI Karnal and also the pot screening at IRA and PAU Ludhiana, and the hydroponic techniques, and we could get a good consensus. And what we basically found in this was, it is not the amount of sodium that is not allowed to get into root, but basically it is the amount of sodium that is prevented from growing into shoot that gave tolerant. That is, low sodium is to potassium shoot ratio played a major role and had a greater correlation. And the establishment was certainly related to some of the transcriptomic networks that we did. We actually started moving with looking at an axiom SNP array to tag this marker trait associations, and we could identify a large number of QTLs basically on the linkage group 3 and linkage group 6, which we published in MDPI Molecular Sciences. And what we started looking is, can we identify what are the genes and the gene regulatory networks that were actually playing? As I said, that necessarily, while almost all the traits decreased in the stressed genotypes, whether it was days to flowering, whether it was days to maturity, or the relative water content, or the membrane stability index, basically, the stress tolerant genotypes were able to have lesser cellular damage, and therefore lesser leachates were released. And the susceptible genotype, the leachate analysis indicated that they had greater cellular damage, and as a result, the ECE of such solutions when grown in stressed conditions was very, very high. Now, when we look at the ionic dynamics, that was what I was speaking of. So there are differential gene expressional analysis. Almost we saw about 50% of the genes, of the 21K genes, differentially expressed genes, which were either upregulated or downregulated along with their bin values. And that's what we was looking, that we had certain co-responsive segregating genes. And when we did a gene ontology study, we found that mostly they belong to three groups. There were the membrane factor binding proteins, or they were the cellular component entities, roughly equal percent, and to some percent they are the cellular processes, whether they were the response to stimuli or the metabolic processes. So what we were interested were the cellular components and the molecular components, which we thought that would play a major role. And then when we did the heat map and clustering of the most 50 salt responsive differentially expressed genes, both in the susceptible as well as the tolerant, over the different permutation and combinations, we saw that a major role was played in calcium transporting, whether it was ATPase gene or the CDPKs, the aquaporin group, or the HAG genes, which played. And we also looked at the families, whether it was the NAC family, MIB, or the ERF. As I said, if you remember in my drought, where we were validating these genes in the heterologous system, those heterologous plants, the transformed Arabidopsis, also proved good amount of tolerance not only to drought but also for heat and also for salinity. And we could publish a paper of the HSPs using the ERF family. So these were the the four major families group of the genes that were upregulated. When we started looking at the transcription factors, which were either upregulated or downregulated, we had the similar trend with respect to the DIGs. And as I said, they basically belong to the important regulators of salinity stress. Now, when we started analyzing the metabolic pathways that were triggered, we have seen that the energy rich pathways were basically triggered, followed by those of the secondary metabolites that were involved in phenylpropanoids or isoflavonoids. And we could come out with how these DEGs played a role under salinity, which we published in a high impact factor EEB, while we see that the signal sensing genes worked through aquaporins, preventing the exclusive extrusion of sodium with respect to potassium. So the transporter genes played a major role here. So while the hormone regulation gene looked at the gene expression, which looked at the ionic balance or the cellular integrity or the osmotic protectant, but basically it was the transporter genes which prevented their intrusion into the root, or prevented their intrusion from the root to the shoot. In the root, if they were invariably coming in some of the tolerant genotypes, we had certain tonoplast ATPase and PPase phosphorylase genes, which lead to their vacuolization through compartmentization. So invariably, as I was saying, almost all the tolerant lines had very low sodium is to potassium ratio. And this is what we could actually identify in our genome-wide transcriptome analysis, which was published in EB, which I said. We actually submitted a large library, Dr. Neeraj and our group, into the NCBI database. And then started now looking at how to mobilize these particular lines. which we could also publish in our Nature Genetics, where the resequencing of these 429 lines, including the genomic accession, could not only provide a idea of domestication and agronomic traits, but a plethora of genes for these biotic and abiotic stresses. Now, I won't go deep into this, but I would like to dwell my last few slides into the new breeding cycle, which you, as young persons who are going to use these techniques to understand. Already from marker assisted breeding to identifying novel alleles, genomic assisted breeding is moving towards haplotype breeding and genomic selection. Basically, most of these breeding concepts and plans have evolved what we call from human and animal genetics, where we try to increase the breeding value of the population per se. That is why IQ over years among the generations is becoming more better, because parents are choosing for their children or children are choosing for themselves elite into elite crosses. And that's what we do in case of pedigree breeding of sires. You take the best stallion and find the best mare, which has almost 7 to 8 pedigrees for the previous generations, whose pedigree is known, record is maintained, the value of the progeny is tested by the breeder, and then these are crossed. So what you basically are trying to do, increase the intensity of the genes for the selected trait or group of traits, so that the breeding value of the succeeding generation is better than the base generation. This is what we call as genetic gain, which we used to do phenotypically, right? Maybe it's not long time before that, like today people used to look at astrology. Tomorrow, maybe your generation, your next to next generation will have a genomic chip. I remember Dr. Varshne, within 10,000 rupees, developed a whole genomic chip to the agricultural god, Professor M.S. Swaminathan, and has gifted me. And now this genomic chip you can make in 2,000 to 3,000. Maybe we'll have that will say, okay, my genomic chip, when some girl asks for my great grandson, they will ask, what is the genomic chip of your great grandson? Yes, no diabetic prone genes, no asthmatic prone genes. Yes, he has genes for high IQ, maybe expressed, maybe not expressed. He has resistance genes. Yes, this matches. It's not a head friend. That's what we are going to do with cheaper genotyping costs, with making selections of target years, maintaining favorable alleles, taking more generations through speed breeding. Hickey and his group from Australia have already indicated this. So what we are trying to do in case of plant breeding is, where we had to take almost 15 years with the advent of marker assisted breeding selection, we have almost halved this. So now if we can take more than 3 generations, this is with 2 generations, I can do it in 6 years. But then I can't handle a large base population. You know, the availability of variability is the basis of plant breeding. If there is no availability of variability within the available 5 fingers, or within the available of 5 lines, I'll be making a choice. But then I will maintain this because it takes time to evaluate them. Somewhere on BC to 4, you see the funnel narrowing. But with this genomic selection using rapid generation advancement, I can have a large amount of fixed lines, and I can very rapidly narrow them into, by the second year itself, into a narrower funnel, which I am doing in the 4th year here. So therefore, I can handle such large mouthed funnels, because I know that after second year, just using genomic selection, I can actually handle lesser. So this is also cost is to benefit ratio. You need to understand why private universities, why foreign universities are scaling high, because they're looking at quality controls, they're looking at cost is to benefit ratio, and that needs to be sieved into the private and the public partnerships and the public breeding program and the educational systems, that there needs to be a TQM, whether you're breeding a bull or whether you're developing a variety. So the genomic selection aids greatly. With this of the rapid generation advancement, we have started already genomic selection. We are very proud to say that in India, We are the first, and also we are only third in the world to have deployed genomic selection in case of chickpea. It takes a huge amount of problem, first of all, to fit these models. We could fit the models and check the accuracy, which we of course published in Frontiers in Plant Science. We had hiccups because we started using the Kabuli to predict the Desi. Then we did understand with these difficulties, and we could find that Desi could better predict for various characters the Kabuli, but not vice versa except for the seed yield. But when we looked at the regression with respect to the phenotypic evaluation with that of the genotypic evaluation, the genomic selection, we found that the genomic selection predicted similarly or much more better than the phenotypic prediction with greater selection efficiencies. Of course, initially we had problems as we used the dark perform or the now we have moved down to agri genome SNP array, almost 2K SNP array. So we have started moving. In brief, I would like to say that the next generation genomic approaches are very useful to develop climate change ready crops. This is not that one crop, one solution for everything. This is only a technique that would increase the precision breeding, like we started using the regression analysis in 1940 and the ANOVA of Sir Ronald A. Fisher. As we move to the PCR techniques, the genomic selection techniques are here per se. The haplotype breeding techniques are here per se. You need to source good phenotyping from the germplasm collection. If your phenotyping is poor, you'll be going for molecular phenotyping, generating gels of pictures on many PCRs, but nothing would come out. You can always make correlations. These are called as nonsense correlations because that is just a statistical package. I know some of my colleagues who have divided drought as susceptible and resistant, categorized them, and made it a single gene inherited. If it was so easy, today almost all the lines in the world would have been drought tolerant. But no, so it is not so. You need to work out for each crop with precision phenotyping technologies and the genotyping selection strategies then would become a success. I'm thankful to Director IRI, my head, and most importantly, my whole group of the students, including Dr. Neeraj, who has joined as my scientific colleague, all my SRFs, whether it is Sneha, Uma, we had Rajesh, the technical officer, and Dr. Chauhan, Apurva, Nilesh, my doctoral student, Sneha, who is currently doing with the University of Western Australia, Supriya, then we have Uma Shankar, Jorbain, we have Dhanakumara, another student here. Then to all my collaborators from Indian Institute of Pulse Research, National Institute of Plant Biotechnology, PIU, Kota Preeti from Sihor, Dr. Yasin RLB CAU, Dr. Chaturvedi, my friend, philosopher, and guide, who has greatly guided in these programs, from Sabor, Nandyal, Dharwad. All my funding agencies, this is just not possible without them, whether it was ICAR, my mother institute, IRI, or the DAG, DBT, and the NTPC, my international funding agencies of Australia, India, Generation Challenge Program, and the Indo Canadian Pulse Genomics Initiative, and my national funding programs of DBT, DST, and the National Fund programs. My international partners, Dr. Rajiv Varshne and Dr. Manish Rurkiwal, who have now shifted to Murdo and Khalifa Center for Biotechnology. From I.S.G.B.R.D, Dr. Tapan, my student who was here, later joined I.S.G.B.R.D, Dr. Shiv Agrawal and Aladdin Hamovi, Dr. Professor Kadambod Siddiq from UWA, Australia, EAB Summit, who was earlier in Cornell, Dr. Liz Punaramu and the group, Nicholas, now also from NIBR, Saskatchewan, Canada, and UCI, UK. Thank you. I'm open to any questions.
You are able to hear me, sir?
Yeah, I'm able to
Yeah, I'm
hear
able to
and
hear and
thank
We
you,
would like
Arun.
thank
to
you, Arun.
Yeah, we would like to really thank you for such so extensive presentation which you have shown over here. We have students also over here. I would like to ask one question before I put the floor to students and other participants. The question is that you work so hard in developing any of these varieties like the chickpea and other things. So I would like to know that for exactly how much the time these varieties are there, for a longer period of time. Like,
Sir,
okay,
let me put it.
let me put it.
This is a
This is
nascent
a nascent
program.
program.
Only
Only
last
last
year they went
year
into
they
the
went into
breeding
the breeding
chain.
chain. Mhm.
So when they go
So
into the
when they go
breeding
into the
chain, it
breeding
takes
chain, it
almost
takes
2 years
almost
or
two
three
years
years
or
to
three
reach
years
to
to
the
reach to the
farmers.
farmers.
But not to
But
miss
not
on
to
it,
miss on
we
it,
have given
we
out
have given
large
out
F&D
large F&D
demonstration classes
demonstration
last
classes
year,
last year,
and they are now
and they are
occupying
now
almost
occupying almost
8% in the
8%
central
in the
India,
central India,
particularly Pusa
particularly
Chitti
Kusach
Manav.
in Manav.
And
And
I'll give you
I'll give you
sometimes, for example,
sometimes, for
there are
example, there are
farmers
farmers
like
like
Lodi
Lodi
from Narsipur area,
Nursinpur area,
Inde, who are
India,
taking
who
out
are taking out
4 tons per
4 tons
hectare,
per hectare,
which is
which is
equivalent to a
equivalent
wheat
to a
crop in
wheat
the tea
crop in
area.
the area.
This
Mhm.
is the
This is the
smallest thing. They're
smallest thing.
new,
They're new.
they're just getting
They're
into
just
the
getting
breeding.
into the beginning. And also, sir, in these varieties, can the new traits be added after a period of time, say after 5 years or 10 years, down
That's
the
good
road? That's good
option, sir.
option,
We
sir.
are not
We
waiting
are
for
not
four or
waiting for
five
4 or
years.
5
We are now
years. We
only
are now
itself
only
starting
itself
out.
starting out.
For example,
For
in
example,
Pusa
in
Chitti Manav,
Kusachiki Manav,
we are
we are
pyramiding the
pyramided the
ascochyta blight
ascochyta
and the
blight
watering
and the
with
water
rainfall
rainfall
tutorials.
tutorials.
We have started
We have
that.
started
That's
that.
what I was
That's what
speaking.
I
We
was
are now
speaking.
going
We are
into
now
a
going into a
convergence program.
convergence program.
Not only that,
Not only
all the
that,
mega
all the
varieties
mega
that have
varieties
been
that
released.
have been released.
A mega
A
variety is
mega
a
variety is
line
a
that has
line that has
almost more
almost
than
more than
400 quintals
400 quintals
breeder seed
breeder
production.
seed production.
So now we
So
are
now we are
mobilizing these
utilizing these
QTLs into these
QTLs into
mega
these mega
varieties.
varieties.
That was what I
That
was
was what
saying.
I was saying.
GT11 has
JT11 has
800 quintals of
800
breeder
quintals
seed
of breeder
production.
seed production.
So
Mhm.
when I
So when
mobilize
I
the
mobilize
QTLs
the
into that,
QTLs into that,
it will immediately go into the farming system because it becomes a EDV.
And sir, one more question, since here we have a lot of students and for their information that there are qualitative traits and there are other quantitative traits. So in general, when you try to do the improved variety, so by which way you start it in general?
Sir, there are
Sir,
certain
there are
things,
certain things,
the
the
qualitative
quality
traits like the grain color, the protein content,
content,
these need to
these
be
and
picked
the
upon.
resistance, like resistance to
The
diseases. The
wilt and
wilt
tolerance
tolerance.
is a must. If you have to release in the central zone, wilt plus ascochyta blight is a must if you want to release northwest plain zone. So these, while the breeding program are inbuilt, and we need to take care. For example, I develop a drought tolerant variety, but among these 8 locations that it has been tested, if it shows wilt susceptibility in any of the 2 locations, it is not released. It becomes an academia.
Sir, I would also like to request you to tell you briefly, since I have seen your video where Prime Minister, our Honorable Prime Minister, have visited your fields where you have developed the varieties. If you can tell you in briefly about to our all the participants and students, I will share
Friends,
the video with
uh,
them.
we are very,
We
very
are very, very
happy
happy.
that even it's not just that that it has been continuing, that our collaborations with either NCBI or with UWA have been long been recognized, and they have been mentioned in the presidential address. Apart from it, the varieties that have been developed have been released by PMO. They have been dedicated to the nation in the PMO uh, in the last two releases by the Prime Minister's office. Not only that, he has specifically visited these advanced breeding lines when he visited the ICRISAT and greatly appreciated the work and the leadership that the breeders of India brought in legume breeding in particular and chickpea breeding in the world. Now we are recognized as world leaders in case of legume breeding in the world.
So, is there any other questions from any students and other participants? Yes, sir. Okay, any questions? Anyone would like to ask any questions? So, if there are no questions, I would again like to say thank you for your time. And I'm really sorry that we have to postpone your talk because our
Not at all. It's always a
participants.
pleasure, sir.
Yeah. Okay, thanks a
And
lot,
I
sir.
and I thank you
And
for the opportunity.
and your talk is, sir, online on YouTube, and our students would be watching it again and again.
Thank
Very
you, sir. Thank you,
informative.
sir. Thank you.
Okay, sir.
So I'll stop share. I need to log out, but I don't know from where I log out.
Yes, sir.
Sir, we will remove you accordingly. You may please continue with your works and turn off your camera. We have with our second presentation, that is a student's presentation. We have Mr. Rahul Singh, student of MTech Biotechnology from RPS Engineering Technical Campus. I would request Rahul Singh to please come up on the stage and start with this presentation. The topic of the presentation is in silico analysis of non-tuberculosis mycobacterium, abscessus, NTM, granuloma proteins.
ये लगाना होगा इसमें खोल ले तो इसी में आ जाएगा इसको ऐसे घुमा
Good afternoon, everyone. My name is Rahul Singh. I am from Raja Balwant Singh Engineering Technical Campus, Srinagar. From there, I am pursuing Tech and Biotechnology. Today, I am presenting my topic in silico analysis of non-tuberculous bacteria, granuloma protein. Topic to be covered are objective, introduction, methodology, results, and conclusion. Come on to the topic. Our objective is to find out the protein that are involved in the granuloma formation of microbacterium abscess by a computational approach. Introduction. The first thing first, what is bioinformatics? Bioinformatics is defined as the application of tools, computational analysis to capture and interpretation of biological data. It is an interdisciplinary field which harnesses computer science, mathematics, physics, and biology. Now, proteome. The proteome is the entire set of protein that can be expressed by gene cell, tissue, organism at a certain time. It is the set of expressed protein in a given type of cell or organism. Now, what is granuloma? It is a small 0.5 to 2.0 mm collection of modified macrophages or epithelioid cell usually surrounded by a rim of lymphocytes. You can define granuloma as a chronic inflammatory reaction containing a preponderance of cells that the monocyte series arranged in a compact masses. It consists of a microscopic aggregation of macrophages that are transferred into epithelial-like cells surrounded by a core of mononuclear leukocyte, principally lymphocyte and occasionally plasma cells. Basically, granuloma is a tiny area of infection containing a cluster of immune cells, especially macrophages, responding against enduring inflammation. Granuloma formation is the body response to separate the foreign body to nodules in the case when the immune system fails to eliminate. In the early stage of infection, the cell-dependent immunity limits the microbacterial growth in granuloma. Adaptive immunity also play a very important vital role in the limitation of granuloma by the implementation of B and T cells at the infection site. At the time of granuloma maturation, activation of dendritic and other cells like natural killer cells, along with some cytokines and leukocytes, including human necrosis factors and which further recruit more immune cells. These are the pictures of the granuloma formation by the Mycobacterium tuberculosis. Now, Mycobacterium abscess, which is classified into a NTM species rather than MTB complex. Mycobacterium abscess complex are group of rapidly growing multi-resistant non-tuberculous mycobacteria that are responsible for a wide spectrum of skin, soft tissue disease, central nervous system, ocular and other infections. Mycobacterium abscess complex is one of the non-tuberculous mycobacterium species which is present in nature such as soil, natural and drinking water and remain alive even after several water treatment processes. Now, Mycobacterium abscess further classified into three subspecies in 2006, 11 and 13, which are Mycobacterium abscess, Mycobacterium bacillans and Mycobacterium volatilum. And this picture shows the infection caused by the Mycobacterium abscess in first image. You can see the lessons on the face, and in the B image, secretion of the pus from the nodes created by the Mycobacterium abscess in the forearms. And C image shows the post cosmetic surgery appear on the upper eyelids. The methodology I have used in our studies, ortho is the first software which is freely available offline software used for the similarity between the genomes between species of different kinds. And another mega software which is also a offline software is to create a calendrogram and phylogenetic trees. And NCBI is the National Center for Biotechnology Information, which is also free web server and contain more database including nucleotide databases. And OrthoVent2 is a web platform for comparison and annotation of orthologous gene cluster among multiple species. Unicode KB is a freely accessible database of protein sequence and functional information, many entries being derived from genome sequencing projects. String is one another molecular basic software. String is basically a biological database and web resource of known and predicted protein-protein interaction. Here is a collection of database dealing with genome biological pathway, disease, drugs, and chemical substances. These are the species which I have included in my study of Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium premium paratuberculosis. These are the known wet lab group identified granuloma forming species. And another three are Mycobacterium abscus, Mycobacterium volati, and Mycobacterium bacillans, which are NTM subspecies.
So I would like to ask you a question. What is the genome size of these different species? Is it almost the same genome size?
So almost the genome size of the same because they belongs to the bacilli family, but then they classified on the NTM species or Mycobacterium complex species.
So what is the genome size? How much MB?
So 2.5 to 4.6 MB something.
Okay. And you can see the details of all these things in.
Yes, sir. Like outline software is used for the comparison of genomic similarity. So this table represents the genomic similarity between all the three six species which I have included. So the main species in Mycobacterium abscus show the percentile similarity between MTB is 71.78 and 71.79 with bovis and 72.98 with Mycobacterium avium paratuberculosis, which is the highest among these three. Now this diagram shows this is the parallelogram, which is also known as the phylogenetic tree. The first one is based on the genomic similarity, and the B diagram based on the proteomic similarity. So orthologous clustering. So to find out the orthologous protein which are commonly present, or we can say the shape between all the six species, so I create the orthologous protein map. So in my finding, these are the 5 proteins which I have identified, which have the respective Unicode ID and protein identities like the non-reductant protein, putative membrane protein, monomer coenzyme acyl carrier protein, putative polysaccharides, beta acyl synthetases, and S-adenyl S-methone dependent methyl transferase. These are some proteins and also some are enzymes and their respective domain status, string formation, H and K phosphate. Now this chart shows the string interaction database, which is done by the string database. So the protein which I have found in these 5 proteins which I have found in my study, to validate the result, I have constructed the protein-protein interaction map with the MTB granular proteins which are already wet lab identified, such as the RDMO locus, which is responsible for the cell death and secretion of cytokines from the infected macrophages, and E.S.G.B.R.D system, which is also virulence vector present in the MTB. By removing this E.S.G.B.R.D system, the virulence vector of the MTB is laid down, and another one is intra-macrophage separation system. So, conclusion, to summarize my study, I have to like to say, using an in silico analysis of non-tuberculous mycobacterium capsicus bromide proteins, done in silico functional characterization and validation with experimental MTV granuloma proteins for the strength of finding that proposed protein might play some role in amnioxicus granuloma. So our study provides some useful insight and can serve as a basis for further studies on microorganism proteins, and also it will help in the finding and creating a drug targeting mechanism and creating a particular drug.
So the granuloma protein which you have identified, so these are the set of proteins or?
So these are some single proteins basically, because my study is based on the NTM, which is a microbacterium vesicus. So the study is on information about the granuloma fungi or the other pathological mechanism. So not much information available. To further distinguish between the MTM complex and MTB complexes and NTM are so much not clear.
So exactly how you make sure that these are the specific proteins which are responsible
So that's why the protein. So these are the 5 orthologous proteins which are shared between the MTB complex, like movies and avian paratuberculosis. These are already identified granuloma proteins. So these 5 proteins, I consider the protein interaction map with this bovis and avian paratuberculosis. So the interaction, so there are some relation between these proteins. So we can say that these proteins also play somehow involved in the granuloma formation. But we can prove with another way, like gene knockout method, to eliminate some of this protein, that they are, by eliminating this protein, then granuloma formation is occurring or not.
Okay. Nice.
Thank you, sir.
Any other questions? So do you want to answer?
So, when it is not a correction or something, but I would like to just give one quick question. When Dr. Arun asked you a question, what was the question which Dr. Arun asked?
Yes, sir.
When Arun sir asked you a question, what was that question which he asked?
सर ने क्या क्वेश्चन पूछा था आपने अभी प्रोटीन के बारे में?
Sir said that how can I validate the protein, like
First one.
फर्स्ट क्वेश्चन
the data of genome, how many size of this?
These are group of proteins or what? Right? And what was the answer?
So there are some specific particular
Yeah,
proteins.
then you went to the bacteria and then you went to something else, right? So just one small feedback, it's not a correction. Whenever somebody asks a question, whether you are in an interview or you are in your pages in Viva or anything, it is always good to be very, very precise and never dwell to any other. That will increase by your success multiple when you are answering the questions. And then those people on the next side of the dais will be always highly educated and more, they will be knowing more than us. Like sir will be knowing more than us what we are thinking. Knowledge on the subject, he will have more. So if we remain very precise, he may not be able to think another question and put you into this. Thanks.
Thank you, Mr. Rahul. Next in sequence, we have Mr. Tulsidas Vishwas from the same college, RBS Technical Campus.
Good afternoon all. I am Tulsidas Vishwas from Raja Balwant Singh Engineering Technical Campus. I am here to present my topic on fermentative production of beta-galactosidase by using mutative strain of microorganisms. Introduction. Beta-galactosidase, commonly known as lactase, is widely used in food industry to manufacture lactose hydrolyzed dairy products. It hydrolyzes lactose into glucose and galactose. It is found in various biological systems, namely plants or microorganisms. However, yeast, molds, and bacteria still remain the only source for commercial purpose. Beta-galactosidase is used in food industry to improve the sweetness, solubility, flavor, digestibility of the dairy products. Beta-galactosidase is also important in conversion of cheese waste from the dairy industry into various useful products. Lactose. Beta-galactosidase is the enzyme responsible for breaking down lactose into our body. During childhood, beta-galactosidase enzyme is found in the guts in large quantity, but enzyme production decreases significantly as a person gets older. Due to beta-galactosidase enzyme deficiency, many people struggles from digesting the lactose found in milk. This leads to the problem of lactose intolerance. In India, 65% of people are lactose intolerant and 70% around the world. Sources of beta-galactosidase. Beta-galactosidase are found in plants, animals, and in microorganisms. Beta-galactosidase in plants play a role in plant growth, lactose hydrolysis, and fruit ripening. Microorganisms most widely used for the production of the enzyme Sclerosis lactis, Sclerosis fragilis, Sclerosis marcianus, and Aspergillus niger. Genus Sclerosis produces intracellular enzyme, while genus Aspergillus produces extracellular enzyme. Beta-galactosidase production. Aspergillus niger extracted from dairy waste and mutated it to produce beta-galactosidase. I used LPCB staining method to detect Aspergillus niger and examine the stain microscopically at 100x to observe the microbe. The production of beta-galactosidase was done by submerged fermentation method. Mutation of the microorganism. Aspergillus niger has been placed under ultraviolet light for 2 minutes at different distance from the light at 10 cm, 15 cm, and 20 cm to mutate it at various levels. Optimized parameter for beta-galactosidase production. I optimized 3 parameters for the production of beta-galactosidase enzyme: temperature, pH, and incubation period. Temperature optimization. To study the effect of temperature on beta-galactosidase production, the cultivations are carried under different temperature levels, 20 degrees centigrade to 40 degrees centigrade on the rotatory second. In this, at 35 degrees
If you wish, I can resend the mail, sir. If you wish, I can resend the mail.
activity was observed. pH optimization.
Sorry, sir.
Usually the effect
On a
of
WhatsApp.
computer latest with the production, but the East culture, the cultivation was carried under different
Yes, sir.
use
I'll
level
resend it on a
from
WhatsApp.
pH
I'm
5
resending
to
the mail
9
as well, sir.
on the rotatory shaker. In this, the reader
Yes, sir.
adds pH study, the
It's
maximum meter
arthur.whitaker
electricity data
at the rate
was observed.
icloud.com.
Effect of incubation period. To find out the optimal incubation time for the return
Right, sir.
electricity
Right, sir.
activity, the fermentative medium was inoculated for 120 hours and samples were drawn at 24 hours time interval from 24 hours of
इनकी फ्लाइट है
incubation.
तो ये सर्टिफिकेट और ऑनर इन्हें करना है
किसकी
At,
फ्लाइट है?
ये सब
at 96 hours
potential amount of phytochemicals that are present in the plant samples, and they can be utilized to formulate the modern form of drugs. Although some of them show potential result in inhibiting strains like Mycobacterium smegmatis, the Mycobacterium smegmatis was used due to its non-pathogenicity and ease of handling. However, the result for different strains of Mycobacterium can be further tested upon and it is a matter of further studies. Several plant species like bubbles have very promising results against several microbes. The study can further be carried forward for the extraction of phytochemicals and formulation of modern medicines. Thank you.
Any questions from anyone?
Yes, sir.
methanol को क्यों यूज किया आपने?
So methanol was used because the extraction procedure it was previously used for different studies. So we are using methanol.
No, why you use a different profile? You have like one is extremely hydrophilic, other is extremely dark. All the ones which we use. So what is the purpose of that? You can use water, but
Yes.
you are using ethanol, then you have the chloroform, then you have the N-hexane, which is the most hydrophobic. So what is the reason for this thing?
So the extraction with the use of ethanol and methanol, it is quite more.
So in another way, I can ask you the question that these compounds which are there, what is the degree of their hydrophobicity or hydrophilicity? What do you understand by this thing? There are certain things which can be dissolved in water.
Yes, sir.
Another can be dissolved in organic solvents.
That is hydroponic.
So any other questions from any? Thank you.
Thank you very much, Mr. Keshav Singh. Next in the sequence, we have special invited lead speaker from Madhya Pradesh, Dr. I.S.G.B.R.D sir, District Fluorosis Officer from Government of Madhya Pradesh, Dhar. Sir is having the teaching and research experience of more than 10 years and is presently working effectively on fluorosis. I request sir to please come upon the stage and present his work. Also, I would request Honorable Dr. R.K. Kulsresh sir to please co-chair this session. Sir will be co-chairing this technical session.
Good evening, everybody. Myself, Dr. I.S.G.B.R.D, District Fluorosis Consultant, धार. Actually, मैं मध्य प्रदेश के 4 डिस्ट्रिक्ट में फ्लोरोसिस डिसीज के ऊपर वर्क कर रहा हूँ और मैं आपको एक ऐसी बीमारी के बारे में बताना चाहता हूँ जो एक बहुत ज़्यादा मात्रा में लोगों को हो रही है. इसका रीजन है मैन फ्लोराइड. जी आपके अभी मैंने देखा है, यहाँ पर भी मैंने देखा है बहुत सारे बच्चों को डेंटल फ्लोरोसिस के सिम्टम्स मैंने यहाँ पर देखा है. तो इस तरीके से हम बचने के लिए कुछ प्रिवेंशन ट्रीटमेंट और सारी चीज़ें आपको मैं शेयर करूँगा. टॉपिक साइड इफेक्ट एंड फ्लोराइड बैटल एंड प्रिवेंशन कंट्रोल ऑफ फ्लोरोसिस. फ्लोरोसिस क्या है? शरीर में फ्लोराइड की अधिक मात्रा हार्ट तथा सॉफ्ट टिश्यू में संग्रहित होने से फ्लोरोसिस रोग होता है, जो कि अस्थियों में विकृति तथा दांतों में विवरण डिस्कलरेशन के रूप में होता है. फ्लोरोसिस निवारण नियंत्रण कार्यक्रम अंतर्गत स्वास्थ्य मंत्रालय भारत सरकार के द्वारा 19 राज्यों के 156 जिलों में फ्लोरोसिस से बचाव एवं नियंत्रण को अभियान के रूप में चलाया जा रहा है. कार्य योजना बिंदु लोक स्वास्थ्य यांत्रिकी विभाग से फ्लोराइड प्रभावित ग्रामों की जानकारी एकत्रित कर कार्यक्रम को प्रारंभ किया गया. फ्लोरोसिस प्रभावित क्षेत्रों में बीमारी से पीड़ित व्यक्तियों को उपचार और परामर्श, फ्लोरोसिस के निदान तथा उपचार की क्षमता वृद्धि. कार्यक्रम का उद्देश्य देश में फ्लोरोसिस का नियंत्रण एवं बचाव. हमारे यहाँ पर इंडिया में फ्लोराइड की कितनी ज्यादा कौन सा स्टेट अफेक्टेड है? 70 to 100% प्रभावित जिले आंध्र प्रदेश, गुजरात, राजस्थान सबसे ज्यादा फ्लोराइड अफेक्टेड स्टेट है. 40 to 70% में हमारा उत्तर प्रदेश भी शामिल है. 10 to 40% अफेक्टेड जिले असम, जम्मू कश्मीर, केरल, छत्तीसगढ़ तथा बंगाल के प्रभावित.
ये सर जमीन में जो फ्लोराइड है उस वजह से है या?
हाँ सर, वो सारी चीजें मैं बहुत सारी डिटेल में बहुत अच्छे से शेयर करूंगा, आपको पूरा डिटेल बताऊंगा किस जमीन में फ्लोराइड की वजह से भी होता है. कुछ हमारा दैनिक जो आहार होता है उससे भी हमें एज ए फ्लोराइड मिलता है, जैसे काला नमक हो गया, काली चाय हो गई, लेमन टी हो गया, बहुत सारी चीजें, चिप्स, कुरकुरे, ये क्या बोलते हैं भेल पूरी, बहुत सारी चीजों का उपयोग करने से हमें फ्लोराइड मिलता है. ये हमारे दैनिक जीवन, इसकी वजह से भी लोगों के बच्चों के दांत अफेक्टेड हो रहे हैं. मैं बहुत सारी चीजों डिटेल और ट्रीटमेंट एंड प्रिवेंशन सारी चीजें शेयर करूंगा. सॉरी. इस तरीके से जो आंध्र प्रदेश के स्टेट हैं, आंध्र प्रदेश में 9 डिस्ट्रिक्ट फ्लोराइड से प्रभावित हैं. असम में 6, बिहार में 11, छत्तीसगढ़ में 5, गुजरात में 4, हरियाणा में 2 और जम्मू कश्मीर में 1 और झारखंड में 13 और कर्नाटक में 19 और केरला में 2, मध्य प्रदेश में 15 डिस्ट्रिक्ट में से 5 डिस्ट्रिक्ट में मैं भी एज गवर्नमेंट ऑफ एमपी के अकॉर्डिंग वर्क कर रहा हूँ. और जो जितना प्रिवेंशन हो सकता है मैं गवर्नमेंट से मिलकर, गवर्नमेंट ऑफ इंडिया, गवर्नमेंट ऑफ एमपी से मिलकर के काम कर रहा हूँ. और महाराष्ट्र की बात करें, महाराष्ट्र में 7, राजस्थान में 30. इसके बाद मैं आपका उड़ीसा की बात करूं, उड़ीसा में 3 डिस्ट्रिक्ट, पंजाब में 3 और तमिलनाडु में 1, तेलंगाना में 9 और उत्तर प्रदेश में 10 डिस्ट्रिक्ट फ्लोराइड से प्रभावित है, जिसमें आपकी जस्ट बाजू का लगा हुआ मथुरा में भी हमारा प्रोग्राम गवर्नमेंट ऑफ इंडिया से चल रहा है. राष्ट्रीय फ्लोरोसिस निवारण नियंत्रण कार्यक्रम के अंतर्गत डॉक्टर विवेक वर्मा है, वो इस पे वर्क कर रहे हैं गवर्नमेंट ऑफ यूपी की तरफ से. तो उसमें क्या बोलते हैं, इस सारे प्रोग्राम में किस तरीके से बहुत काफी विलेज अफेक्टेड है मथुरा में. अभी मैंने कई बच्चों को देखे तो मैंने देखा कि मतलब फ्लोरोसिस की ज्यादा यहाँ पर भी कुछ समस्या है. तो मैंने उपाध्याय सर से भी बोला और सब लोगों से बताया कि यहाँ पर फ्लोरोसिस की समस्या है, तो इसको किस तरीके से बच सकते. तो मैंने कहा स्पेशल मेरे लेक्चर लूंगा, इससे कुछ प्रिवेंशन ट्रीटमेंट हो सकता है. इस तरीके से मैप के द्वारा बताया गया है. हाईली अफेक्टेड स्टेट में राजस्थान, गुजरात, आंध्र प्रदेश और 40 से 70% में आपको उत्तर प्रदेश भी शामिल है. ये फ्लोराइड किट होती है, एफटी किट फील्ड टेस्टिंग किट होती है। इस फील्ड टेस्टिंग किट के माध्यम से जिस तरीके से हमारा मेडिकल मैनेजमेंट में ये हीमोग्लोबिन का टेस्ट करते हैं, जो हमारी एएनएम होती हैं, वो गांव एएनएम आशा होती है, वो घरों में सर्वे करने के लिए कोई दस्तक अभियान या कोई स्वास्थ्य शिविर होता है तो वहाँ जाती हैं, तो ब्लड सैंपलिंग करती हैं। इसी के बाद ब्लड सैंपलिंग करती हैं तो उसमें देखते हैं कितना हीमोग्लोबिन है। इसी तरीके से पीएचई विभाग के पास फील्ड टेस्टिंग किट होती है। इस फील्ड टेस्टिंग किट से पता करते हैं कितना पीपीएम पानी में फ्लोराइड है। इसमें मार्किंग के द्वारा अपन मेजर कर सकते हैं, लेकिन ये 100% इससे मान नहीं सकते कि इसमें इतना फ्लोराइड है। इसके बाद हम लैब में भेजते हैं। लैब में भेजने के बाद देखते हैं, सॉरी, इसमें मार्किंग के बाद अगर पानी का कलर पिंक आ रहा है तो मान के चलो फ्लोराइड नहीं है, उसमें अच्छा है, नॉर्मल है, 1 ppm से कम है। वो अगर येलो आता है तो मान के चलिए उसमें फ्लोराइड है। फिर उसके बाद मेजर करने के लिए हम लैब में पानी को सैंपलिंग करते हैं और टेस्टिंग करते हैं। इसके बाद हमारा आयन मीटर जो लैब स्टैब्लिश है, जो हमारे यहाँ मध्य प्रदेश में करीबन 15 डिस्ट्रिक्ट में से 8-10 डिस्ट्रिक्ट में लैब स्टैब्लिश है। ये धार में ये मेरी लैब है, यहाँ पर हमारा टेक्नीशियन है, ये सब टेस्ट करते हैं। इसमें जो रिएजेंट होते हैं, तेजाब, थूथली और इस तरीके से बहुत सारे रिएजेंट होते हैं जिनका उपयोग करके हम टेस्टिंग करते हैं। इसमें ड्रिंकिंग वाटर में 1 ppm से ज़्यादा अगर फ्लोराइड आ रहा है तो पानी पीने लायक नहीं है, लेकिन हम दैनिक जीवन में उसका इस्तेमाल कर सकते हैं। दूसरी बात, अगर उसके बाद हम यूरिन सैंपल लेते हैं। यूरिन सैंपल का कलेक्शन करने के बाद अगर 1 ppm उसमें निकल रहा है ज़्यादा, तो मान के चले कहीं ना कहीं वो व्यक्ति फ्लोराइड वाला पानी पी रहा है, फ्लोराइड उसके खान पान में ज़्यादा आ रहा है। तो उसके बाद अगर उसमें 1 ppm से ज़्यादा फ्लोराइड निकल रहा है तो उसको काउंसलिंग करेंगे और जो पानी को इस्तेमाल कर रहा है उसकी भी टेस्टिंग करवाते हैं हम। टेस्टिंग करवाने के बाद देखते हैं अगर उसमें फ्लोराइड लेवल कम आता है तो उसको जो दैनिक जीवन जो आहार ले रहा है उसके बारे में डिस्कशन करते हैं।
इसका सर जनरली जो ग्राउंड वाटर होता है उसमें कितना होता है? 1 से 3 तो आपने बता दिया, बट मैक्सिमम कितना आ जाए?
तो सर एक्चुअली इसमें कोई लिमिट नहीं, कहीं तो 20-20, 25-25 ppm फ्लोराइड मिलता है, 15-15, 20-20 ppm। अभी आपको पता है कितने सेंट्रल फ्लोरोसिस केस हो रहे हैं आपके हरियाणा में भी है, और बहुत सारी जगह पे ऐसे केसेस देखने को मिलेंगे आपको। केस में कॉम्प्लिकेशन डिफॉर्मिटी आ जाती है। और ये इस तरीके से हम अभी आपको मैं हालांकि बताना चाहता हूँ कि हमारी लैब जब पानी की टेस्ट करने के लिए भेजते हैं तो सारे ऑर्डर होते हैं, 17 प्रकार के टेस्ट होते हैं ये पानी की। लेकिन क्या होता है, हमारे लोग कोई भी होता है वो पानी का टेस्ट नहीं करवाता जो हम ट्यूबवेल्स लगाते हैं। आज कंपलसरी हो गया है लोगों को अपने घर में, जो अगर अपना स्वास्थ्य अच्छा रखना है तो पानी का टेस्ट करें। चाहे आप आरओ लगाएं तो आरओ पानी से आप बोले आपके आरओ के द्वारा कितना कौन कौन से एलिमेंट आप रिमूव कर रहे हैं, कौन कौन से एलिमेंट आपके पास आ रहा है पानी में, वो कौन कौन से एलिमेंट की हमें आवश्यकता है बॉडी के लिए, ये हमें बहुत जरूरी है। जिस तरीके से मैंने 2 सैंपल टेस्ट करवाया था फ्लोराइड को लेकर के, स्पेशल मैं फ्लोराइड के ऊपर फोकस करता हूँ। तो इसमें 13 नंबर पे 2 हैंडपंप का सैंपल लिया था मैंने, बाकायदा उसमें केमिस्ट्री साइन भी है। 2 हैंडपंप पे एक हैंडपंप में 5.01 फ्लोराइड निकला है, एक हैंडपंप में 0.48 फ्लोराइड निकला हुआ है। इस तरीके से हमें अपने पानी की जो घर में आजकल क्या हो गया है कि सभी लोगों को सेपरेट मकान, सभी लोगों को सेपरेट मकान बन रहे हैं और सभी लोग होल ट्यूबवेल्स का इस्तेमाल से पानी कर रहे हैं। तो हमें पानी का टेस्टिंग के बारे में बहुत ज्यादा ध्यान देने की आवश्यकता है। नेक्स्ट, फ्लोरोसिस के प्रति जागरूकता क्यों आवश्यक है? फ्लोरोसिस पर मेडिकल कॉलेज में बहुत कम बताया जाता है तथा इस पर शोध बहुत कम होती है। फ्लोरोसिस गंभीर विकलांगता जो कि अपरिवर्तनशील तथा उपयुक्त उपचार न होने वाली स्वास्थ्य संबंधी समस्या है। अगर किसी व्यक्ति को एक बार ये डिफॉर्मिटी हो गई तो व्यक्ति सही नहीं हो सकता प्रॉपर कंडीशन में, लेकिन हम इस बीमारी को आगे बढ़ने से रोक सकते हैं। फ्लोरोसिस का शारीरिक विकास धीमा पड़ जाता है। शहरी क्षेत्रों में फ्लोरोसिस का प्रभाव दिन प्रतिदिन बढ़ता जा रहा है। शहरी क्षेत्रों में इसका फ्लोरोसिस डिसीज क्यों बढ़ती जा रही है? क्योंकि हम बहुत सारी चीज़ों का जैसे जिन चीज़ों का हमें इस्तेमाल नहीं करना है और हमारे बच्चे बहुत इस्तेमाल करते हैं, मैं वो चीजें भी आपको बताऊंगा। फ्लोरोसिस का बचाव तथा नियंत्रण कार्यक्रम पूर्ण रूप से गतिशील नहीं हुआ। फ्लोरोसिस का जनसंख्या में आयोडीन का भी परिलक्षित हो रही है, क्योंकि फ्लोरोसिस का आयोडीन का शरीर में शोषण नहीं हो पाता। फ्लोरोसिस का पॉपुलेशन में एनीमिया के केसेस भी बढ़ते जा रहे हैं। तो मैं एक चीज और बताना चाहता हूँ, सभी लोग जानते होंगे आयोडीन का हमारी बॉडी के लिए कितना बड़ा रोल है। हमारी बॉडी के लिए जो नमक होता है उसमें आयोडीन होना बहुत जरूरी है। आयोडीन नहीं अगर नमक में रहेगा तो हमारा ब्रेन का डेवलपमेंट नहीं होगा। इसलिए गवर्नमेंट ऑफ इंडिया ने आयोडीन एक डिफेंसिव प्रोग्राम भी स्पेशल चलाया है लोगों को कि आयोडीन वाला ही नमक का इस्तेमाल करना है। आपने देखा होगा पहले हमारे गांव में ये वो ढेला वाला नमक होता था ना, उसका इस्तेमाल करते थे। जो नहीं जानवरों को, हाँ हाँ ढेला वाला नमक, हालांकि उसने गवर्नमेंट ने अभी बैन कर दिया है, वो नमक कहीं पर भी नहीं मिल सकता है, वो अपन जानवरों को भी नहीं खिला सकते। नेक्स्ट, फ्लोरोसिस की सुरक्षित मात्रा सामान्यतः पीने वाले पानी में 1 ppm अर्थात इससे अधिक पाई जाती है तो पानी पीने योग्य नहीं है। वातावरण के कारण क्षेत्र का तापमान अधिक होना, कम वर्षा, कम मात्रा, जमीन में फ्लोराइड की चट्टानें, फल सब्जियां अधिक फ्लोराइड वाले क्षेत्रों से आना, फ्लोराइड टूथपेस्ट का उपयोग किया जाना विशेषकर बच्चों में। दो चीजें यहाँ मैं बताना चाहता हूँ कि फल सब्जियों से भी हमें फ्लोराइड मिलता है। जिस तरीके से कोई हैंडपंप कोई खेत है, खेत के आसपास कोई ट्यूबवेल लगे उससे सिंचाई कर रहे हैं, उस सिंचाई के द्वारा हालांकि देखा होगा आपने कहीं हमारे यहाँ पर नहर सरफेस वाटर का पानी अच्छा होता है, नदी तालाब कुएं का पानी अच्छा होता है। लेकिन मैं आपको बताना चाहता हूँ जैसे राजस्थान हो गया, हमारे मध्य प्रदेश कुछ एरिया में नदी तालाब की थोड़ी नहीं है, कुछ पहाड़ियों में रह रहे हैं तो वहाँ पर हैंडपंप या ट्यूबवेल से ही पानी इस्तेमाल करते हैं, उससे भी हमें क्या बोलते हैं फ्लोराइड की दिक्कत होती है। फ्लोराइड टूथपेस्ट का उपयोग किया जाना विशेषकर बच्चों में। अब बात आती है फ्लोराइड के बाद। फ्लोराइड के बाद फ्लोरोसिस 3 प्रकार की बीमारी होती है, जिसमें डेंटल फ्लोरोसिस, स्टेंटल फ्लोरोसिस, नॉन स्टेंटल फ्लोरोसिस। अधिक फ्लोराइड की मात्रा वाले क्षेत्रों में रहने वाले दांतों में के बच्चों में विवर्तता दिखाई देती है। शुरुआती अवस्था में साफ चमकदार सफेद दांत धीरे धीरे हल्के रंग के हो जाते तथा उन पर सफेद पीले स्पॉट उभरने लगते हैं। धीरे धीरे स्पॉट का रंग गहरा हो जाता है और धारियों के रूप में दांतों के किनारों पर दिखने लगते हैं। लेट स्टेज पर पूरा दांत काले भूरे रंग के हो जाते तथा वो विच्छेद होने लगते तथा दांत धीरे धीरे गलने लगते हैं। अधिक फ्लोराइड प्रभावित क्षेत्रों के लोगों के दांत उम्र से पहले गिर जाते हैं और दंतहीन हो जाते हैं। इस तरीके से ये अभी यहाँ पर और आगे नेक्स्ट होता है। ये आप लोग इस तरीके से पिक देखने को मिल रही है, लोगों के टीथ इस तरीके से डेवलप हो रहे हैं। गहरी माइंड में जरा सा स्पॉट देखने को मिलेगा आपको, माइल्ड में आपको थोड़ी और डेवलप मिलेगा, मॉडरेट में और ज्यादा आएगा, सीवियर में इस तरीके से केसेस। सीवियर में उन बच्चों के ज्यादा केसेस ऐसे देखने को मिलेंगे जो बच्चे गांव के हैं। गांव का मतलब है उनको प्रॉपर तरीके से न्यूट्रिशन नहीं मिल पाता है, क्योंकि उनको फल सब्जियां प्रॉपर तरीके से उनको पानी और सादा खाना में इस्तेमाल करते हैं, तो उनको क्या बोलते हैं न्यूट्रिशन की चीजें नहीं मिल पाती हैं, जिससे उनको इस बीमारी से और ज्यादा घेर लेती है। नेक्स्ट, ये देखो आपको बिल्कुल पिक्चर क्लियर दिख रही होगी, इस तरीके से टीथ बच्चों में देखने को मिलते हैं। ये हम कितना ही डेंटल सर्जन के पास अगर इस तरीके से बच्चे टीथों में प्रॉब्लम दिख रही है, हम दुनिया के कोई भी डेंटल सर्जन के पास जाएंगे, अगर बोलेंगे कि हमारा ये इस तरीके से टीथ सही करवा दीजिए तो नहीं कर सकता है, क्योंकि इंटरनली प्रॉब्लम है। ऐसा नहीं है कि वो ऊपर से हो सकता है। कैप लगा सकते हैं डेंटल सर्जन, इसके अलावा और कुछ नहीं कर सकते। कैप हमारा कोई ट्रीटमेंट या प्रिवेंशन नहीं है। हमारा प्रिवेंशन यही है कि आप फ्लोराइड वाली चीजों से बचें। नेक्स्ट, इसके बाद डेंटल फ्लोरोसिस है, डेंटल कैरिज। डेंटल कैरिज भी हमें होती है, अगर पानी में फ्लोराइड बिल्कुल नहीं है तो हमें डेंटल कैरिज भी हो जाती है, तो हमें पानी में फ्लोराइड की हमें जरूरत है। नेक्स्ट, इस तरीके से पूरे टीथ डिस्टर्ब हो जाते हैं। नेक्स्ट, ये पिक्चर मैं आपको जब मैं फील्ड में जाता हूँ मैं खुद देखता हूँ बच्चों को, आपको देख रहे हो बच्चों के टीथ इस तरीके से फ्लोरोसिस के लक्षण हैं। नेक्स्ट, इस तरीके से बच्चे, आपने देखा हो कितना बच्चा को कुपोषित लग रहा है, पूरी बॉडी में फ्लोराइड की वजह से पूरे दांत खराब होना, ये तो बहुत लेट स्टेज में होता है, उसके पहले वो कुपोषित हो जाता है, बॉडी पूरी ड्राइनेस हो जाती है। नेक्स्ट, इस तरीके से उसके बाद हम इस गांव की बात सर्वे करने के बाद मैं और ह्यूमन राइट्स कमीशन में पेशी पे गए हुए थे, क्या फैसिलिटी हो रही है। कलेक्टर साहब थे वहाँ, जिस डिस्ट्रिक्ट की मैं भी था, और PHE। तो हम लोग इसको मतलब बहुत लोग बाग हल्के में लेते हैं। इसका रीजन क्या है, क्योंकि इसकी वजह से, फ्लोराइड की वजह से किसी व्यक्ति की डेथ नहीं होती। नेक्स्ट, इसके बाद स्केलेटल फ्लोरोसिस की बात करें। स्केलेटल फ्लोरोसिस में क्या क्या सिम्टम्स आते हैं? हील पेन, पेनफुल रेजिस्टेंस, जॉइंट मूवमेंट, डिफॉर्मिटी लिम्स, बैकएक, इन एक्सट्रीम केसेस पैरालिसिस, मस्कुलर डिस्ट्रेस, प्रीमेचर एजिंग। नेक्स्ट, तीन जो स्केलेटल फ्लोरोसिस की हम आइडेंटिफिकेशन करने की 3 एक्टिविटी कराते हैं, जो हमारे हेल्थ वर्कर हैं वो करते हैं। इस तरह की 3 एक्टिविटी जो हाईली अफेक्टेड एरिया में ये एक्टिविटी करा करके चिन्हित करते हैं और उनको ट्रीटमेंट के लिए प्रिस्क्राइब करते हैं। नेक्स्ट, अब आपको मैं बताना चाहता हूँ, स्केलेटल फ्लोरोसिस इस तरीके से डिफॉर्मिटी आ जाती है, ये बच्चे इस तरीके से उनकी डिफॉर्मिटी आ जाती है। नेक्स्ट, पूरा प्रॉपर तरीके से देखने को मिलेगा। ये देखो, आपने देखा होगा पूरे पैर इस तरीके से डिफॉर्मिटी से मुड़ रहे हैं। नेक्स्ट, ये आपको बिल्कुल हॉस्पिटल में हम लोग पेशेंटों को ले गए थे उनको स्क्रीनिंग करवाने के लिए, ट्रीटमेंट कराने के लिए। नेक्स्ट,
यहाँ सर कितनी लेवल कितना है? यहाँ 5 1 से ऊपर है या 5 तक है?
अभी एक्चुअली सर मैं अभी कुछ नहीं, कहाँ पर, इस जगह की, इस जगह पहले प्रॉब्लम थी, यहाँ पर 5 के आसपास ppm था।
मतलब जो ये डिफॉर्मिटी,
हाँ, डिफॉर्मिटी 5 के आसपास ppm था, और जो गांव के बच्चे थे, ट्राइबल माहौल के थे, तो एक्चुअली उनको जो पानी मिला, अभी आप फील्ड में जाएंगे देखने के लिए, बड़ी स्थिति खराब है।
एक सर बात बताइए, अवेयरनेस कितनी है लोगों में?
आ रही है, लोगों में आ रही है, लेकिन अवेयरनेस प्रॉपर तरीके से नहीं आ रही है। मैं कह रहा हूँ ना, इसका दूसरी बात, एक चीज और बताना चाहता हूँ सर, कि जो फ्लोराइड है, जो पानी है, उसका कोई कलर नहीं होता है। अगर पानी का कोई कलर हो, फ्लोराइड वाला, जैसे लाल, पीला, हरा या कुछ तरीके से, तो उस पानी का अवेयरनेस आता, इसका बिल्कुल एकदम सॉल्वेंट रहता है।
आपकी जो ये किट है, ये आप लोगों में डिस्ट्रीब्यूट
डिस्ट्रीब्यूट ये वाली किट PHE विभाग करती है, ये वाली किट PHE विभाग करती है। हमारा काम है ट्रीटमेंट, हम हेल्थ डिपार्टमेंट से, हम लोग हेल्थ डिपार्टमेंट को डिफॉर्मिटी आती है तो उनको ट्रीटमेंट के रूप में, प्रिवेंशन के रूप में प्रिस्क्राइब करते हैं। जी, नहीं, PHE मतलब पब्लिक, वो PHE लोक स्वास्थ्य यांत्रिकी विभाग, गवर्नमेंट का, पब्लिक नहीं, पब्लिक है तो लोक स्वास्थ्य यांत्रिकी विभाग, PHED बोलते हैं उसको। नेक्स्ट, ये सॉरी, एक बैठो। इस तरीके से हम लोगों ने एक ऐसा, ये आपको पिक्चर दिख रही है, हैदराबाद की है, कर्नाटका की, वहां पर पूरा हम लोग ट्रेनिंग पे गए हुए थे, पूरा का पूरा गांव मतलब कोई एक्टिविटी नहीं कर पा रहे, दादा बैठे हुए हैं, ये अपने मतलब कोई दैनिक एक्टिविटी करने के लिए रस्सी पकड़ के काम करते हैं। एक पेशेंट नहीं, वहां पर कई पेशेंट, बहुत ज्यादा डिफॉर्मिटी है। ये हमारा एक बच्चा था, इस बच्चे की हमने ट्रीटमेंट करवाया है, सर्जरी करवाई है, आज से रिकंस्ट्रक्टिव सर्जरी करवाई हुई है, जो इरीजारो सर्जरी होती है रूसेट टेक्निक से, जो मध्य प्रदेश में, इंडिया में इस तरीके से पहली सर्जरी हमने करवाई हुई है, जो सक्सेसफुल हो गई है। इस तरीके से आप ये बच्चे की, मैं खुद फीडबैक लेने के लिए जाता था और देखते थे, और आज ये बच्चा प्रॉपर तरीके से उसका पैर सीधा हो गया है, एकदम डिफॉर्मिटी, जो एक्चुअली फ्री ऑफ कॉस्ट में हमने राष्ट्रीय फ्लोरोसिस निवारण कार्यक्रम के तहत की हुई है। नेक्स्ट, मैं बराबर जब भी हर इस पेशेंट के लिए, हर मतलब अपना घर का पेशेंट समझ के उसको हर 3 दिन में फीडबैक लेने के लिए, MI हॉस्पिटल इंदौर में है, बहुत बड़ा हॉस्पिटल है गवर्नमेंट का, वहाँ पर हम लोग जाते थे फीडबैक लेने के लिए, प्रॉपर ट्रीटमेंट की किस तरीके से कैसा क्या चल रहा, सारे डॉक्टर्स आ जाते थे मेरे को बताते, डॉक्टर सर इस तरीके से हमारा ये एक्चुअली इम्प्लांट है, इस इम्प्लांट में आप पैर को किसी भी तरीके से मूवमेंट कर सकते हैं। इसको आप जो ऑर्थोपेडिक सर्जन होते हैं वो कंप्यूटर में फिट कर देते हैं, किस पेशेंट की किस ट्रीटमेंट में किस तरीके कितना टाइम लगता है, तो उसमें उसके अकॉर्डिंग फिट हो जाती है। ये करीबन 1 महीने में प्रॉपर तरीके से पेशेंट फिट हो गया था। नेक्स्ट, ये देखिए, आपको एक पैर की हमने सर्जरी कराई हुई थी, पैर पूरा एकदम सीधा लग रहा है, दूसरा पैर अभी मुड़ा हुआ है। इस पैर इसको सीधा होने के बाद इसके 3 इंच लंबाई भी बढ़ गई थी, फिर हमने दूसरा एक उसको शूज कैलिपर्स बनवाए हुए थे। नेक्स्ट, पेशेंट हैप्पी, डॉक्टर हैप्पी एंड डिफॉर्मिटी करेक्टेड। नेक्स्ट, ये फिर वहाँ फीडबैक लेने के लिए मैं जाता था, अभी भी जाता हूँ। तो इस तरीके से वहाँ की उसके घर की भी पिक्चर है, प्रॉपर तरीके से फिट हो गया। ये वहाँ की आशा बहन जी जिन्होंने हेल्प की, आशा आंगनवाड़ी नेक्स्ट, फिर हमने सक्सेसफुली स्टोरी बनाई थी जो मीडिया में दी हुई थी। नेक्स्ट, इस तरीके से नई दुनिया, सारे जो स्टैंडर्ड पेपर, सभी पेपर में हमने न्यूज दी हुई थी। नेक्स्ट, ये तरीके से हम एक्टिविटी। अब बात आती है नॉन स्केलेटल फ्लोरोसिस। नॉन स्केलेटल फ्लोरोसिस एक्चुअली हमें दिखता नहीं है, इंटरनली हमें प्रॉब्लम बनी रहती है। जैसे हमें क्या होता है, लूज ऑफ एपेटाइट, न्यूशिया, पेन इन स्टोमच, कॉन्स्टिपेशन, इंटरमिटेंट डायरिया एंड ब्लोटिंग स्टोमच फ्लूइड। मतलब ये समस्या अगर हमें कंटिन्यू बनी रहती है तो कहीं ना कहीं पानी का गड़बड़ होता है। नेक्स्ट, इसके बाद हमारी ये समस्या आती है तो हमारी इंटेस्टाइन भी हमारी डिस्टर्ब होती है। नेक्स्ट, हमारे इंटेस्टाइन 100% जब न्यूट्रिशन होती है तो अंदर जो हिस्टोपैथोलॉजिकल हमने वर्क किया, देखा, इस तरीके से आप इंटेस्टाइन दिखती है। नेक्स्ट, जब 20% मींस न्यूट्रिशन मिलता है तो इस तरीके से इंटेस्टाइन इंटरनली डिस्टर्ब होती है। नेक्स्ट, ये 3% में पूरी तरीके से हमारे इंटेस्टाइन डिस्टर्ब हो जाती है और हमें कॉन्स्टिपेशन, कब्जियत, दस्त, पेट फूलना, ये समस्या बनी रहती है कि कहीं ना कहीं हमें फ्लोराइड का रीजन हो सकता है। मैं ये नहीं कह सकता हूँ कि ये हो सकता है, उसके लिए ट्रीटमेंट के लिए हम उनकी यूरिन का टेस्ट करें, उसके ब्लड का सैंपलिंग करते हैं, तो उसके बाद पता चलता है। नेक्स्ट, ये हमारी जो मसल्स होती है, मसल्स में आपने फर्स्ट पिक्चर नॉर्मल मसल्स ऊपर देखी होगी, आपको बिल्कुल क्लियर दिख रही है। नीचे फ्लोराइड मसल्स में पूरी मसल्स डिस्टर्ब हो जाती है, जिस वजह से हमारी इनको डिफॉर्मिटी आ जाती है। नेक्स्ट, ये हमारी आरबीसी, इसमें घबराहट, बेचैनी, बहुत सारी समस्याएं होती हैं। इसमें आरबीसी भी क्या क्या प्रॉब्लम है, वीकनेस, फैटिग, लो हीमोग्लोबिन, डिप्रेशन, ये सारी चीजें डिस्टर्ब होती है फ्लोराइड की वजह से। जहाँ पर हाईली फ्लोराइड अफेक्टेड एरिया है, इस तरह के केसेस बहुत मिलते हैं। नेक्स्ट, इसमें ये हमारा स्पर्म में भी डिफेक्ट होता है। नॉर्मल पूरा स्पर्म मतलब नॉर्मल स्पर्म आप सब लोग जानते हैं, साइंस के स्टूडेंट हैं, और इस तरीके से हमारा स्पर्म भी डिस्टर्ब हो जाता है। जो इस तरीके से डिस्टर्ब होने के बाद स्पर्म ओवम को फर्टिलाइज, जो अभी पहले बताया था कि इसमें प्रजनन की भी समस्या आती है, तो स्पर्म ओवम को फर्टिलाइज नहीं कर पाएगा। नेक्स्ट, अब बात आती है क्या क्या हम कर सकते हैं। सेफ ड्रिंकिंग वाटर इंटरवेंशन, इंश्योर द फ्लोराइड लेवल डज नॉट एक्सीड द परमिशन लेवल 1 mg पर लीटर, 1 mg से ज्यादा नहीं होना चाहिए। इंश्योर एनकरेजमेंट रेन वाटर हार्वेस्टिंग, अभी आपने देखा होगा गवर्नमेंट ने स्पेशल जितनी भी बिल्डिंग बन रही है, सारी बिल्डिंगों में रेन वाटर हार्वेस्टिंग लगाने के लिए प्रेशर है, अप्रूवल ही नहीं मिलता है। कोई भी अगर बिल्डिंग आप बनाने के लिए जाओगे, उसमें रेन वाटर हार्वेस्टिंग बनाना कंपलसरी कर दिया है। अगर रेन वाटर हार्वेस्टिंग नहीं है तो परमिशन नहीं मिल पाएगी शायद, मेरे हिसाब से। सेफ ड्रिंकिंग वाटर, लेक, पॉन्ड, रिवर, कैनाल, ऊपरी सतह, सरफेस सतह की जितना भी पानी होता है वो अच्छा होता है, लेकिन उसको हम डायरेक्ट नहीं पी सकते। उसको हम क्या बोलते हैं, क्लोरिनेशन करके ही पानी का इस्तेमाल करें, चाहे वो नदी का हो या तालाब का हो, क्योंकि बहुत सारा केमिकल उसमें बहुत सारी चीजें डाल देते हैं, जिससे हम क्या बोलते हैं दिक्कत आती है। इसको हम क्लोरिनेशन करके ही इस्तेमाल करें। अब नहीं, रेन वाटर जो नीचे चला जाता है, रेन वाटर हार्वेस्टिंग होता है ना सर, देखा होगा, जो जैसे अपने बिल्डिंग है, हाँ नीचे चला जाता है वो। एक्चुअली वाटर लेवल बढ़ाने के लिए रेन वाटर हार्वेस्टिंग वो नीचे चला जाता है। एक ऐसा बिल्डिंग के बगल से पाइप डाल देते हैं। हाँ, फ्लोराइड वाटर लेवल बढ़ेगा ना सर। एक्चुअली फ्लोराइड की चट्टानें नीचे पाई जाती हैं, जितना नीचे अपन गहराई में 400-500 फीट के नीचे पानी निकालेंगे, हो सकता है आपके यहाँ पर 150-200 फीट पे आ जाता हो, 150, क्योंकि आपके बाजू में जमुना नदी है, तो हो सकता है उसकी वजह से। लेकिन आप सोचिए, जस्ट बाजू में मथुरा है। हमारा जो राष्ट्रीय फ्लोरोसिस निवारण नियंत्रण कार्य गवर्नमेंट ऑफ इंडिया के द्वारा जो प्रोग्राम चल रहा है, तो आप सोचिए मथुरा में चल रहा है, इसलिए प्रॉब्लम तो होगी वहां पर, डेफॉर्मिटी अच्छी खासी होगी, तब ये प्रोग्राम इम्प्लीमेंटेशन हो रहा है, नहीं तो इम्प्लीमेंटेशन की कोई जरूरत नहीं होती। इसलिए इस चीज को अवेयरनेस होने की बहुत जरूरत है। अब बात आती है इसको कैसे हम सपोज दैट कहीं पर 2 ppm वाला फ्लोराइड वाला पानी है, कोई अल्टरनेट सोर्स नहीं है, तो हम क्या करें? फ्लोराइड जैसे ही हमारी बॉडी में पहुंचता है, हमारी बॉडी से कैल्शियम रिमूव होते हैं। कैल्शियम रिमूव होगा, हड्डियां हमारी कैल्शियम की बनी होती है, टीथ हमारे कैल्शियम के बने होते हैं, तो हमें इसको लिए प्रिवेंशन के रूप में कैल्शियम, विटामिन सी, विटामिन ई एंड एंटीऑक्सीडेंट, इन सब चीजों का इस्तेमाल करना है। अब बात आती है ये सारी मेडिसिन हम कब तक खाएंगे? ज्यादा मेडिसिन खाएंगे तो भी हमें बॉडी में प्रॉब्लम क्रिएट हो जाएगी। इसके लिए हम हरी सब्जियों का इस्तेमाल करें। नेक्स्ट, सप्लीमेंटेशन के रूप में हम इन सब चीजों का दूध पिए, पालक खाएं, और आंवला में आपको पता है सबसे ज्यादा सब विटामिन सी होता है, और गांव का हर एक व्यक्ति जानता है, बहुत इजी गोद में मिल जाता है। अगर हम विटामिन सी आंवला से लें तो हमारी बॉडी की इम्यूनिटी भी अच्छी डेवलप होगी, जिससे हमारे कभी भी कोई बहुत सारी फ्लोरोसिस बीमारी तो ठीक है, कोई भी बीमारी नहीं हो सकती है, जो हमें रोग प्रतिरोधक क्षमता भी बढ़ाने का काम करेगी। नेक्स्ट, सप्लीमेंटेशन एंटीनेटल डो में जो हमारी गर्भवती माता है उनके ऊपर भी बहुत ज्यादा जरूरत है फोकस करने की, तो हमारा स्वास्थ्य विभाग करता है उसमें। नेक्स्ट, इस तरीके से हमारे प्रोग्राम के द्वारा आइडेंटिफिकेशन ऑफ फ्लोराइड अफेक्टेड विलेजेस, डेवलपमेंट, डायग्नोसिस, फैसिलिटी, सेंसिटाइजेशन, वर्कआउट, ट्रेनिंग, ICBC, सप्लीमेंटेशन, कैल्शियम, विटामिन सी, एलॉन्ग विद डाइट काउंसलिंग, ये हमारे इस प्रोग्राम के द्वारा किया जाता है। नेक्स्ट, अब बात करें क्या करें क्या ना करें, डू एंड डोंट्स। कैल्शियम रिच फूड में मिल्क प्रोडक्ट, ग्रीन लीफी वेजिटेबल, विटामिन सी रिच फूड, सिट्रस फूड, आयरन रिच फूड, ग्रीन लीफी वेजिटेबल, बनाना, गुआवा, ब्रिंजल। क्या नहीं करना है हमें? कंटिन्यू ब्लैक टी, लेमन टी, ब्लैक लॉबस्टर, टोबैको, सफारी, फ्लोराइड टूथपेस्ट, इन सब चीजों का इस्तेमाल ज्यादा हमें नहीं करना है। नेक्स्ट, इस तरीके से आपको एक पॉइंट और आपको देखने को मिल रहा होगा, ये रेड मार्क अगर किसी हैंडपंप पे लगा हुआ है, मान के चलो वो हैंडपंप सूख गया है, उसमें पानी नहीं आ रहा है, लेकिन अगर उस हैंडपंप में येलो निशान लगा है तो इसका मतलब है पानी, उस हैंडपंप में फ्लोराइड वाला पानी आ रहा है। दूसरी बात, एक्चुअली गवर्नमेंट ऑफ इंडिया ने बोला है कि इस तरीके से जितने हैंडपंप हो जिसमें फ्लोराइड आ रहा है, सारे हैंडपंपों को बिल्कुल कैप करना है। लेकिन क्या होता है, गांव की स्थिति बदतर होती है, वहां पर कोई अल्टरनेट सोर्स नहीं होता है, जिस कारण से गांव के लोग उस हैंडपंप को बंद नहीं करने देते। वो कहते हैं हम इस पानी का इस्तेमाल नहीं करेंगे, लेकिन हम दैनिक जीवन में इस्तेमाल कर सकते हैं। तो इस तरीके से, लेकिन क्या होता है, कई बच्चे जो पहचानते नहीं है और पानी का इस्तेमाल कर लेते हैं, और कंटिन्यू, जो गांव के अनपढ़ होते हैं, कुछ कम पढ़े लिखे लोग होते हैं, तो इस पानी का इस्तेमाल भी कर लेते हैं। लेकिन हम उसको मार्किंग करवाते हैं, पीएचई विभाग के ऊपर प्रेशर डाल के कि आप मार्किंग करिए, कलेक्टर साहब को बोलते हैं, वो इस तरीके से करवाते हैं। और येलो ब्लू निशान, इसका मतलब अच्छा पानी है। नेक्स्ट, इन सब आपको पता है द सीक्रेट और बिहाइंड द फ्लोरोसिस प्रिवेंशन, रेनफुल डाइट ईटिंग एंड डाइट काउंसलिंग। इस तरीके से ब्लैक टी, काला नमक, इन सब चीजों का इस्तेमाल हमें ज्यादा नहीं करना, क्योंकि ब्लैक टी एक्चुअली जो चाय होती है उसमें फ्लोराइड रहता है, हमें ब्लैक टी का ज्यादा इस्तेमाल नहीं करना। नेक्स्ट, ये सारी चीजों आपको देखने को मिल रही है। ये इसकी वजह से हमारे शहरी क्षेत्रों में फ्लोराइड की मात्रा, फ्लोरोसिस से लोग, बच्चों के दांत खराब होते हैं। ये सारी चीजें जो कोई गांव का बच्चा नहीं खाते, हमारे शहर के लोग खाते हैं। इन सब चीजों में फ्लोराइड रहता है। इसमें देखने को मिला ब्लैक टी, देखो ब्लैक लॉबस्टर, सैदा नमक, ब्रत का नमक, लोहारी नमक एंड पाकिस्तान नमक, 157 ppm फ्लोराइड रहता है। ये मैं नहीं बोल रहा हूँ, हमारी रिसर्च के द्वारा बताया गया है।
ब्लैक साल्ट जो है,
हाँ,
वो दो तरह से,
जी,
एक सी साल्ट को ब्लैक साल्ट बनाया जाता है, और एक ब्लैक साल्ट रॉक से।
जी।
तो ये जो है आप सी साल्ट को ब्लैक साल्ट जो बनाया उसको रिकमेंड कर रहे हैं या जो रॉक साल्ट को रिकमेंड कर रहे हैं?
नहीं, एक्चुअली ब्लैक रॉक सॉल्ट मतलब मीन्स जो काला नमक होता है जिसमें अपन इस्तेमाल करते हैं।
काला नमक ही दो तरह का, सफेद से काला बनता है, मैक्सिमम जो है।
अब मुझे इसके बारे में नहीं मालूम सर, मुझे इसके बारे में नहीं
सैदा
मालूम।
नमक अलग चीज है। काला नमक सफेद से ही काला बनाते हैं।
एक्चुअली जो बताया गया अब इसके बारे में जो गवर्नमेंट ऑफ इंडिया से जो गाइडलाइन है उसमें बताया गया इस तरीके से इसको यूज नहीं करना है।
सर इस बात को थोड़ा आगे
मैं बिल्कुल आपकी बात पॉइंट नोट कर लूंगा और अभी आपसे इसके बाद में डिस्कशन कर लूंगा। एक्चुअली मैं सारी चीजों को इसलिए बता रहा हूँ, ये सारी चीजें हमारे मतलब हमारे इंडाइजेशन को प्रॉब्लम क्रिएट करती है। आपने देखा जब तीखा चीजें खा लेते हैं तो हमें इंडाइजेशन प्रॉब्लम होती है, तो इसका रीजन ये भी हो सकता है। तो ये मैं अपनी तरफ से नहीं बोल रहा हूँ, ये गवर्नमेंट ऑफ इंडिया से ये सारी चीजें चल रही। किसी भी कंपनी किसी भी प्रोडक्ट के बारे में आप नहीं बता सकते। ये जो हमारा कोई मैं अपनी तरफ से नहीं दे रहा हूँ, सारी चीजों पर रिसर्च किया गया है। नेक्स्ट,
ये जो चीजें हैं ये कितने % जो है आदमी के अंदर इसको पैदा कर रही है?
जी सर उसके ऊपर आप हाँ
पॉइंट है,
आप सही बोल रहे हैं। इसका रीजन ये सर अपन कंटिन्यू प्रॉब्लम क्रिएट करेंगे, कंटिन्यू उस चीजों का इस्तेमाल करेंगे।
जो रेडीमेड है जो
हाँ।
भी आपने पिक्चर दिखाई है,
हाँ, हाँ।
है? तो ये लगभग 70% आज के युग में चल रहा
हाँ
है।
चल बोल,
70%।
आपने देखा होगा कुछ छोटे छोटे बच्चे हो जो अच्छे रिच फैमिली से होते हो, उन बच्चों के टीथ भी नहीं निकल पाते,
हाँ।
बिल्कुल पतले पतले निकल जाते, बड़ी स्थिति टीथ भी नहीं निकल पा रहे या इधर उधर डिस्टर्ब हो रहे। तो ये इसकी वजह से भी होता है, क्योंकि आजकल क्या होता है बच्चा रोता है तो चिप्स कुरकुरे तत्काल में उसको दे देते हैं। तो ये सारी चीजें आजकल ये बहुत ज्यादा चलन में आ रहा है।
अगर ये 70 to 80% है, तो गवर्नमेंट को इसको जो है अच्छे तरीके से चेक आउट
आपको चलो सर इसके बाद आप लेकर जानते हो ना सर, चलो ठीक है सर, आप लेकर जानते हो ना, लिकर आपको पता है तो लिकर कितना साइड इफेक्ट करते हैं, फिर भी ब्लैक में और नंबर 2 में कितना अभी लीगल तो ठीक है, और कितना मतलब हाई रेट में, और एक से एक वैरायटी 4-4 हजार रुपए की एक बोतल मिलती है। तो उसके लिए अपन क्या कर सकते हैं, लेकिन डेफिनेटली अपन प्रिवेंशन के रूप में जो जी ग्रीन टी बेटा आप, मैं कह रहा हूँ टी की बात, ग्रीन टी लेमन टी ये डेली इस्तेमाल करने की चीज नहीं है। आप कर सकते हो, लेकिन आप अपनी हैबिट में डालोगे तो कहीं ना कहीं आपको कई लोग बोलते हैं ग्रीन टी से हमारा डाइजेशन बहुत अच्छा होता है,
वेट कम होता है,
वेट का मतलब बहुत सारी चीजें आती हैं, लेकिन उसके साइड इफेक्ट आप कोई भी चीज ज्यादा लोगे। ये आपको मैं ग्रीन टी लेमन टी ये मैं मतलब बता रहा हूँ, यहाँ से नहीं है, वो गवर्नमेंट ऑफ इंडिया से बाकायदा रिसर्च के द्वारा बताया गया है, और मेडिको रिसर्च जो हमारी नॉन मेडिको रिसर्च में इसमें अप्रूव किया गया है कि फिर इसका ज्यादा इस्तेमाल नहीं करना है। नेक्स्ट, ये हमारे हार्ट बोला वगैरह ये सारी चीजें हमारे लिए साइड इफेक्ट देती हैं। नेक्स्ट, इस तरीके से हमारे कुछ नारे लेखन, पीने के पानी में फ्लोराइड है, जहाँ फ्लोरोसिस, पीने के पानी की जांच कराओ, अपने को फ्लोरोसिस बचाओ, दूध दही हरी सब्जियां खाओ, फ्लोरोसिस से मुक्ति पाओ, पीले दांत हड्डी जाम, यह पानी में फ्लोराइड का काम। नेक्स्ट, इस तरीके से हमारे जो गवर्नमेंट कलेक्टर साहब के बाद साथ मीटिंग करके कोऑर्डिनेशन करती हैं। 4 विभाग होता है जिसमें हेल्थ डिपार्टमेंट, पीएचई डिपार्टमेंट, एजुकेशन डिपार्टमेंट एंड महिला बाल विकास ICDS, 4 डिपार्टमेंट मिलकर के अवेयरनेस करते हैं, क्या क्या कौन डिपार्टमेंट का रोल है, सारे रोल प्ले करते हैं। इस तरीके से हमारी मीटिंग होती है, जिला स्तरीय कोऑर्डिनेशन मीटिंग होती है। पानी में फ्लोराइड की मात्रा कम करने के लिए बेटा इसमें क्या बोलते हैं, हमारे EDF प्लांट होती है, इलेक्ट्रिक डीफ्लोरेशन प्लांट होते हैं, वो एक्चुअली PHE विभाग लगाते हैं, वो एक्चुअली काफी कॉस्टली होता है। अब बात आती है लोग बात आरओ, बात आती है आरओ से फ्लोराइड रिमूव तो होता है, लेकिन आरओ में सारे आपको पता है WHO ने बोला है कि आप आरओ का पानी इस्तेमाल नहीं करिए, क्योंकि आरओ से सारे के सारे मतलब मिनरल्स रिमूव हो जाते हैं, निकल जाते हैं। अब निकल जाएंगे तो वो हो जाएगा क्या, डिस्टिल वाटर, तो उसको भी बोला। तो इस तरीके से हमें पानी की टेस्ट करना। मैंने आपको 2 चीज बोली, जब आप आरओ लगाते हैं तो भी आप आरओ वाले से बोलिए आप हमारे इस पानी के सारे मतलब एलिमेंट टेस्ट करके दीजिए, आपके आरओ से पानी में क्या क्या एलिमेंट आ रहा है। नेक्स्ट, इस तरीके से हमारी मीटिंग होती है, अलग अलग डिस्ट्रिक्ट में होती है, और जो सब HR पर्सन कलेक्टर साहब रहते हैं उसको रिकमेंड मैं ही कर उनको बताता हूँ कि इस तरीके से सर ये समस्या होती है। कलेक्टर साहब जो भी एक्चुअली जिले के बॉस कलेक्टर होते हैं वो सारे डिपार्टमेंट को लेटर जारी करते हैं, जो कार्रवाई विवरण होते हैं किस डिपार्टमेंट को क्या क्या करना है, कितने समय में क्या क्या एक्टिविटी करना है, वो करते हैं। इसको नेक्स्ट, ये हमारी चंडीगढ़ में मीटिंग हुई थी, गवर्नमेंट ऑफ इंडिया के डॉक्टर सक्सेना सर हैं, उनके द्वारा क्या बोलते हैं हम लोग 7 स्टेट की मीटिंग हुई थी चंडीगढ़ में, जो 19 में हुई, उसके बाद अभी होना थी, लेकिन क्या बोलते कोविड के कारण नहीं हो पाई, अभी दिल्ली में होना थी। तो इस तरीके से हम लोग 7 स्टेट की मीटिंग में सारे लोग आए थे, सारे CMHO डिस्ट्रिक्ट ऑफिसर हम लोग गए हुए थे मीटिंग में। नेक्स्ट, इसके बाद हमारे मध्य प्रदेश में 6 डिस्ट्रिक्टों में इकट्ठी साथ में मीटिंग हुई थी, जिसमें 6 डिस्ट्रिक्ट के कलेक्टर सब लोग मिलके, और मैं भी अपनी बात रखता हूँ। नेक्स्ट, इसके बाद थैंक यू। आरोप मैं आपको बता चुका हूँ सर, हम लोग हेल्थ डिपार्टमेंट में, हेल्थ डिपार्टमेंट हम क्या करते हैं, जहाँ पर जो पेशेंट मिलता है उन पेशेंट की डाइट काउंसलिंग करवाते हैं, उनको
क्या किया जाता है
हाँ, वहाँ के लिए वही मैं बोल रहा हूँ, वहाँ के लिए उस पानी की जांच करते हैं, हमारे हेल्थ डिपार्टमेंट में पानी की जांच की मतलब स्पेशली उस पेशेंट की पानी की जांच करते हैं। अगर उसमें 1 ppm से ज़्यादा फ्लोराइड आता है तो PHE विभाग को लेटर लिखते हैं, सारे अब सभी का वर्क डिस्ट्रीब्यूट है, PHE विभाग को लेटर लिखें, PHE विभाग उस जगह का पानी बंद करेंगे या जो भी अल्टरनेट सोर्स होता है वो करते हैं। उसके बाद हमारे जो हम स्वास्थ्य कार्यकर्ता होते हैं उनको क्या बोलते हैं बता दिया जाता है, इन पेशेंटों बच्चों को फोकस करें, उनको कैल्शियम विटामिन C मेडिसिन समय समय पर प्रोवाइड करवाएं और डाइट काउंसलिंग के लिए दें। जी सर।
क्योंकि आपने ये कहा कि नमक में आयोडीन वाला ही नमक आजकल लगा हुआ है।
जी जी।
विदाउट आयोडीन जो नहीं करते।
जी।
पर आजकल डॉक्टर्स के रिकमेंडेशन है कि आयोडीन जो है थायराइड का भी कारण बनता है।
हाँ तो सर उसका लेप
उसका आपने कोई एक्सपर्ट बनाया है कि इससे ज्यादा नहीं?
नहीं, हाँ नहीं, अभी इसके बारे में मैंने अभी जो आपने क्वेश्चन किया ना, मेरे से ऐसा क्वेश्चन किसी ने किया हुआ है, तो मैं इसके बारे में आगे नोट करके बढ़ा दूंगा।
कारण क्या है कि थायराइड की आज जो आयुर्वेद के
बिल्कुल आप बिल्कुल सही बोल रहे हैं। इसमें आपको मान जब किसी को हाइपरटेंशन होता है तो डॉक्टर आयोडीन वाला नमक ना खाने के लिए बोलता है, क्योंकि इसकी हाइपरटेंशन की प्रॉब्लम क्रिएट होती है। हाँ हाँ, तो अब इसकी समस्याएं सभी जगह हैं।
सेंधा तो आप अलाउ कर रहे हैं,
नहीं काला नमक, सेंधा नमक, ब्लड का नमक,
काला नमक ब्लैक साल्ट और सेंधा नमक रॉक साल्ट।
हाँ तो
रॉक साल्ट तो अलाउ है ना?
रॉक साल्ट ले सकते हैं, ठीक है सर। क्या कह रहे हैं? नहीं प्रेशर, मैं प्रेशर में जो
क्यों ले सकते हैं सादा नमक जब उसमें आयोडीन नहीं है तो?
कौन माना? एक मिनट, हाँ नहीं तो नहीं एक मिनट, जब
बहुत कम परसेंटेज है। दूसरी बात ये है
जी
कि एक नॉर्मल वाटर पिए आदमी, एक जो है फिल्टर वाटर पिए,
ठीक है,
तो किससे आदमी ज्यादा टॉयलेट जाएगा?
नॉर्मल मतलब एक नॉर्मल वाटर पिए
और एक फिल्टर पानी पिए तो टॉयलेट नंबर आपको चांस,
वो आदमी की कैपेसिटी के ऊपर डिपेंड है ना, उसका किस तरीके नहीं एक बार सर, फिल्टर किडनी फिल्टर क्या कर वर्किंग उसकी
नॉर्मल सा क्वेश्चन है ये।
आप ठीक बोल रहे हैं, लेकिन कंडीशन होती है उसकी आदमी की बॉडी की,
जिसमें DHS और मिनरल्स आर रिच,
TDS,
वाटर TDS,
हाँ TDS,
स्लो यूरिन प्रोसेस रहेगा,
TDS ज्यादा होगा कुछ इसमें।
यस
बिल्कुल सही है।
और जिसमें और मोस्टली जो नेचुरल वाटर है हैविंग द लॉट ऑफ TDS,
हाँ हाँ
दैट हाई गोइंग टू द यूरिन बाय टाइम टू टाइम,
हाँ कंड ज्यादा हो,
स्लो TSS और क्या कहते हैं वो ज्यादा
TDS
तो कम जाएगा।
TDS
जो फिल्टर वाटर है जैसे RO का पानी है, ऑल न्यूट्रिएंट्स है गायब हो गए,
जी
तो वो आदमी ज्यादा जाएगा, ऐसे समझ।
नहीं हाँ आप बिल्कुल सही मतलब ठीक बोल रहे हैं, लेकिन इसकी चीज आपकी उसकी बॉडी की कैपेसिटी के ऊपर डिपेंड होती है।
Depends upon the capacity, you are right. Water, filtered water and free from nutrient or
सर सर इमली एंड आंवला, हाँ इमली एंड आंवला, बिल्कुल सर, विटामिन सी, इमली इमली सर, बहुत सारी चीजें, मतलब एक एग्जांपल दे, आंवला का एक। ओके सर, थैंक यू।
Thank you very much, sir. There are a few important informations for all the participants. We have a cultural program, a band presentation sharp at 6 in Dr. A.P.J. Abdul Kalam Auditorium. And this is a rock band presentation. A rock band is coming for you guys. Also, we have planned high tea and snacks in the plan. Apart from this, we have planned a trip excursion tour to Agra tomorrow morning in 5 a.m. It's 5 a.m. All are kindly requested, if they are joining in this trip, they must maintain the decorum and no nuisance will be accepted anymore. Also, this is to inform that few girls from S.G.B.R.D and a few delegates more left this premises without information. Hence, all are informed now that if you want to leave the premises, you need to inform your hostel warden if you are staying in the hostels, or the organizers. You need not to contact anyone else. Please make a note of this. One more information: while any of the lead speaker is giving his presentation, we need to maintain the decorum, and please make sure you don't laugh in between in any of the case. Thank you. Now we have Professor Arthur Ledecker, sir, from France. Sir is from Oikos Institute and I.S.G.B.R.D, and sir will be giving his presentation on revisiting agriculture, forestry, and international policies for global and national sustainable development in 2022 after the latest IPCC reports and the Russian-Ukrainian war. Preliminary considerations. Sir will start with the presentations. All are requested to please be seated here only. A few more information will be shared after this presentation. Thank you. One more information, sorry for the interruption. We have started with the parallel poster presentations outside this auditorium hall. If anybody is interested to visit for those posters, may leave. Thank you.
Hello, sir. Yes, sir. Please continue with the
Yeah,
presentation.
that's up to me now.
Yes,
Yeah.
sir. It's over to you, sir.
Good afternoon to everybody. I'm trying to revisit the agriculture and forestry and international policies for global and national and global sustainable development. I mean, it's very important to make a distinction between the two, because every country is interested in national sustainable development. But if we are just interested in national sustainable development, we will never achieve what we aim for stabilize the climate in 2050. And that's why we have to revisit it. And because the IPCC, as I will show you, is policy oriented, but not policy prescriptive. But the limit between the two is very small and difficult. And in practice, the working group 3, of which I'm going to speak, is really too restricted to convey the correct messages for people. So what I would like to during my conference, I'm going to make this presentation and show you why I think there is a need for a complementary report to summary for policymakers from IPCC, at least for working group 3. And I would at the end get your impression and your remarks, because I think it's necessary. So, but you will find if it's useful or not at the end. So thank you for listening carefully and for giving me the advice at the end. So this is the scope of what I'm stressing. First, I will tell you what IPC task for and reports are and main messengers here. So I will go through up to the end to the conclusion that is we need a complementary report to summary for policymaker in at least for the working group 3 for mitigation. Otherwise, we'll never achieve it. So. What is the task of IPCC? It's to provide policymakers with regular scientific assessment on climate change, and this is one thing they do very well, I think. And we have to show the implication of potential and future risk. That's working group 3, 1, and working group 2 is to deal with adaptation. And that's also fairly good. But the working group 3, it's on mitigation option, and only activities of this group are considered here. I'm not speaking of the other groups. So usually in IPCC reports, in each group, over 3 groups, we have a summary for policymaker, and that's sometimes about 10 to 20 pages. This year it's 63 pages. That's the only document that policymakers read usually, and they are translated in several languages, so that line by line, and they are proved during a whole week between scientists and representatives of governments. That means representative of governments can remove what they want to remove, because they if they don't want to things to make something and I think it's too prescriptive, they remove it. So it's from 1,000 page or more, you come to 10 to 15 pages, and in between you have a technical summary, which is also only in English. It's approved. But the full report is in English, the technical summary is in English, and only the summary for policymaker is generally known by journalists and by policymaker. So I don't know if you have read ever one of these reports, but they are quite interesting, useful. So, when we are just speaking about the policy summary for policymaker, not about basic reports, which are fairly good. So this year we had the free summary for policymaker on physical sciences basis, impact and adaptation, vulnerability, and of group, working group 3, mitigation for climate change. And I'm going to concentrate on this. So, but these are our main messages. IPCC full report and SPM are necessary for working group and working 2. They are useful. But national greenhouse gas emission per country and per capita, as well as sectoral emission in working group 3, are insufficient and even misleading for policymakers and journalists, especially for land use. It is also ignored or insufficiently dealt with how to avoid land use change and minimize increase of greenhouse gas emission by optimizing land use efficiency, by producing more food and non-food biomass per hectare. It's also what is also insufficient is the regionalization of mitigation adaptation option according to economic specificities and vulnerability as requested in the Climate Convention. And they don't tell you where to change your diets, which actions should take place, and prioritize between in developed and developing countries to preserve food security as well as the climate. So, IPCC are policy oriented, as I said, but not policy prescriptive. That means they can only publish. Rely on published papers and reviewed papers usually. So consequently, what is conveyed to newspapers is sufficient, and citizen conference, for instance, what we had in France in 2020 is also not really useful, because what they proposed was not associated with cost. so they could make any recommendation. Of course, this would have needed much more time and expertise to do that. And the procedure to either improve or contest the proposal made by such groups, there is no procedure to revise that and to give feedbacks. So, for instance, I will show you that for organic farming, they have wrong messages, but they are like organic farming, but for climate change it's completely misleading. So, and this is not discussed. So, and so the average citizen thinks that this is the case. And then average citizens really don't understand properly what should be done, and where is no, and in that case, there is no change to meet the UN targets and to eradicate hunger by 2030 and net zero greenhouse gas emission by 2050, as asked by the United Nations in 2050. So I'm insisting on that because you had a very interesting presentation during this conference. But you should not restrict your activities to your specific scientific activity. You should also have a look on what is going on more globally, and perhaps because it's important to share it with us, with scientists and decision maker, and because we need a transition toward net zero emission, and this is a long and difficult task. And Yellow Vest in France have demonstrated. You may know that they have that high carbon taxes, which were favored by liberal economists, is no longer an option, and we should aim for ecological planning. But this was not really assessed in the last IPCC report. So what is important is to have equitable burden sharing of climate mitigation adaptation, as suggested in Article 3 of UN Convention, United Nations Convention on Climate Change. And this can only be done outside of IPCC reports, because governments would never allow to have all the information I'm going to give you in a report, in an official report, which is too. So this is, if we go back to UN Convention on Climate Change, that it says in Article 3 that parties should protect the climate system for the benefit of present and future generations. That this is what of humankind. That's what everybody knows, but it's also said that it's on a basis of equity and in accordance with common but differentiated responsibilities and respective capabilities. Accordingly, the developed country parties should take the lead in combating climate change and the adverse effects thereof. The specific need and special circumstances of developing country parties, especially those that are particularly vulnerable to the adverse effect of climate change, and those parties, especially developing countries that would have to be a disproportionate and abnormal burden under the convention, should also be considered. But this is, under IPCC work, is considered to be policy prescriptive. That's why we don't have the whole picture when we just read the IPCC reports. So they said that we have to limit the climate risk. So but these are the projections of what countries are going to have in the future, and this is what what should be done. The reduction in greenhouse gas emission to be able to keep below 2 degrees and be below 1.5 degrees. So there is a huge gaps. And the IPCC can just say there's a huge gap, so they cannot say more. They say that we need at least to divide greenhouse gas emission from fossil fuel by 2030. That's fair. But what now to change in agriculture and forestry and food system, and where? This is not really detailed in summary for policymakers. Here you have greenhouse gas emission per capita, today in various countries. Of course, you see here that in North America, the emission per capita are close to 20 ton of CO2, whereas in Southeast Asia, it's sort of 3 to 4 ton of CO2 per capita. Of course, there's a big difference. And what is in green is due to land use change, emission of CO2 due to land use change. So this is one picture. So, but if you go further and you have the cumulative anthropogenic CO2 emission, you see that it's even more. And North America is responsible for 23%, Europe for 16%, and Eastern Asia but 12, 11. So it goes down. So, and if you go per capita, you will see that it would be much higher. In fact, because this at the present stage, the emissions are calculated on territorial emissions. That means emission, for instance, in India, emissions in France, and emissions in US or China. They are, and so that gives you that amount of emission at about 6.6 ton of CO2 per capita in France. But actually, if you take into account the import and export of food and other products that are exchanged, then it's almost twice as high. That means it's about 10.9 ton of CO2 equivalent per person. Of course, that's not as easy to calculate as the territorial emission. But today, everything is based on this. That means the all emission from developed countries are underestimated because they import that they underestimate, and emission from developed countries are overestimated. So that means it's here you have, because they export, and here also an export. So that's a big change, a big difference. And today we are not dealing with that. So this is what is where we are dealing is. It's agriculture, forestry, and land use. So it's 22% of emissions, but you will see that it's actually less than that. But depending how you take into account the methane, it's even more than that. So and one thing which is not, which is sometimes only in technical reports, but not in summary for policymaker. That means policymakers will not read it or hardly read it. That's mean the emission per country depend largely, and the type of emission depends largely over income per capita in countries. So in low-income countries, the greenhouse gas emission come mainly from agriculture and forestry, and of course in rich countries they are also in emission from agriculture and forestry, but there are a lot of other emissions from buildings, from energy sector, and so on. So there is a big difference, and these are basic emissions for survival emissions. For instance, in Africa, it's necessary to have that. So that's a policymaker. This is not in policymaker. So another important thing is related to that in low-income countries, they have no industries, and they have no income to subsidize adequately agricultural inputs. And this is completely different from developed countries. For instance, in Europe, we have the common agricultural policy, and you will see how much more money they get per hectare than in developing countries. So here, subsidies in most developing countries are less than 2%, except in Malawi, where they are about 70.5%, in Bangladesh, and also in India. So, but in other countries, because they have a lot of other activities, so that means the subsidies for agriculture are fairly low in our countries. And moreover than that, in high-income countries, you have about 20% of subsidies for production, for producer, and for consumers, and everything. So in total, it's about 20%. Whereas in low-income countries, the subsidies are only 4.4%. That's a big difference between this type of countries. So this is never shown in IPC's reports. The subsidy no, actually this is in dollar, but it's about the same. But in the past, in Japan was very high, in the EU also, but in Australia and United States it was very small, the subsidy per hectare. But today in France, for instance, we have a farmer have 340 euro per hectare per year from the common agricultural policy budget. So it's a big difference between developing and developed countries, in particular between Africa and developed and rich countries. So we also had a special summary for policymaker on climate change and land. It was a special report in 2019, but they give only average figures, which are not really useful for decision makers. They say how much you get in general from emissions from net CO2 emission from agriculture and forestry, the COH4 emission and methane emission from agriculture, and so on. But this doesn't tell you where they are high, where they should be reduced, and what should be done. So they are not very useful. And this also, in this same report, they only give the change of average changes of cereal yields, for instance, here, of course. But this, as we will see, is not very important and not very useful. They give also the change in inorganic fertilizer use. But this also is not very important because, as you will see, some countries use very much, too much fertilizer, and other countries too little. So and so on. So that means it's also not useful for decision maker. So there is no indication in this IPCC report on difference in yields. Here you have just yields of wheat in France, cereal yields in OECD countries. They have continuously increased. Latin America, South Asia, except in sub-Saharan Africa, where they have remained very low. So that's something that decision maker in Africa should know, but this is in no report. Another very important thing which decision maker should know, and that's the inputs in sub-Saharan Africa are very low, less than 50 kilo per hectare, sometimes even 10 or 15 kilo per hectare per year. Whereas in other countries, in developed countries, it's between 100 and 250 kilo per hectare. In China, of course, they have sometimes two crops per year, but they use too much fertilizer, and they are decreasing now the amount of nitrogen which is put in fields. And in Egypt, they have two crops, but because the amount of land which is available is really too small, so they have to maximize the yields. So this type of so we have big difference. Let's say if you go from Niger, where you have 1 kilo per hectare, to Egypt in Africa, to where you have 600 kilo per hectare. So there is a big difference, and this is never shown in any IPCC report. So this shows that how the nitrogen inputs in Europe have increased since 1919 and after 1950. But it was like that, and the increase of yield was also because of increase of fertilizer, manure, and mineral fertilizer. So of course, before second oil shock, the quantity of fertilizer that was used was a little bit too high, and so it has decreased after that. But still, you need input to have a good yield. So this is what have happened in developing countries since 1960. That in East Asia, in Pacific, it has increased regularly, and also here. But in sub-Saharan Africa, the input have remained very low. So this is one explanation, and you will see later on what will happen. So here you see that in China, they have decided, because they did use too much fertilizer. They have decreased the fertilizer since the last 2-3 years, and so that's much better than before, and this can be used elsewhere. And so what also should be said in IPC report, which is not clearly stated, is that because of this increase of fertilizer use, the land we have much land has been land use change has been avoided. For instance, we have this is the amount of land which is cultivated for cereals since 1962 up to 2014, and this is the increase this is avoided land use change. It's 1.26 billion hectare. That's about 252 billion tons of CO2. So I go back. So and that's really a huge amount, and this is not really highlighted because this is a type of problem that you don't encounter for fossil fuel with fossil fuel, but with land use issues, this is a very important issue. So, for instance, when you come from forest land to grassland, or from grassland to cropland, the carbon stocks in trees this is an average for temperate regions. Average carbon stock in trees and in soil is decreasing, and compared to this is to emission from fertilizer input, they are very small. Of course, they occur every year, so there is a big change here, and so you better not have land use change. But when here you can make calculation. For instance, for budget per hectare for wheat and rice. Of wheat, in 1915 we had 8 ton of grain per hectare, and emission was 0.6 ton of CO2. In 2000, of course, the emissions are much higher, 2 tons, but the yields are also higher. And if you take into account the additional biomass production, just the straw, not the rest, not even grain, just the straw, you see that you can avoid, by using and converting straw properly into energy, you can avoid 3.6 ton of CO2. That means is more than the increase of CO2 per hectare that you had here. So that means it's compensating more of the emission, the increase of yield. You have also that in I'm not insisting on that, but in India, because in addition to CO2 emission and N2O emission, you have also methane emission, and so if of course this is emission. So, but if you have a two-season drainage and a lot of and a fairly good fertilization, then you also have a better system. Of course, it's less efficient than with wheat because you have methane emissions, but you can have also a better system when not taking into account the increase of emission from the increase of fertilizer. So this is you can. So that means, for instance, we showed that in Malawi, if we could fund about 10 euro per ton of avoided CO2, would have divided the cost of public subsidies by 2. And today people are speaking of 50 euro per ton of CO2. That means it's far less. Even with 20 tons, it would have cost nothing in Malawi to co-fund the inputs to reduce the land use change. So voilà, this is to compare with carbon taxes today. And but of course, some NGO think that this was too much, to 50 kilo per hectare, and they never say anything. This is a North American NGO, World Resource Institute, but they never say anything about the amount of fertilizer which is used per hectare in their countries. So speaking of agricultural policy is not really possible in IPCC reports for summary report for policymaker. So this is one of the reasons we would need an extra report, additional report, because when you get guaranteed price, as in Europe or in US, when you have a control of change and you get subsidies like in all developed countries, and since 1929, the great crisis, the agricultural policies are no longer in developed countries. They are no longer based on liberal exchange. They are always administrated, and so otherwise it would not be possible to have this increase that we had. So and Bill Gates is also for green revolution in Africa, but like the World Bank, he thinks that the governments have nothing to do there, and they have no, we should not interfere. And this is based on Washington consensus. Of course, this kind of discussion cannot take place in the IPCC reports, but they are very important because here, for instance, they had made World Bank made a lot of recommendations which are quite reasonable. But when you come here, you see that if there is no subsidies, and we have to remove all the subsidies for inputs, and that's completely impossible and doesn't exist in any developed countries, and even in developed countries except in Africa. So this is change, but this shows clearly that when you use more fertilizer in some countries, it does reduce both land use change, because here you have two option scenario. You can double the yield, total production. You can double the area of cropland, or you can double the yield up to a certain point, of course. And here is what you avoid, deforestation that you avoid, and these are harvest loss which don't take place here, but they take place here. So if you have all this, that means you have both land use change with fertilizer. You can both reduce land use change and total greenhouse emissions. I insist on that because usually people think that you add more fertilizer, you are going to have more greenhouse gas emissions. So this, in a nutshell, is a comparison between conventional farming and between low or organic farming. And here you have the same production, but because the average yield is half of what is for conventional farming, you need twice as much land. That means you have to remove this part of forest, or this land cannot be preserved for different service, environmental service, and cannot be used also for to produce biomass for energy. That means there's a big difference between the two. But this is ignored, as you will see. This is generally ignored by NGOs advocating organic farming. And here you have increase of what happened in France between 1950 and 2000 with our total production, and that because the yields have increased, we have saved 14.4 million hectares, about a quarter of the French territory. That's quite a lot, and also be reduced emissions during this time. In Africa, the situation is completely different because of deforestation. There's no increase of yields, and they had to deforest. They emit about 1 billion ton of CO2 per year. That's about 1 ton of CO2 per capita. So it's not negligible. And this is because the difference between Asia and Sub-Saharan Africa. In Sub-Saharan Africa, the yields have increased, and okay, and here you see that in Sub-Saharan Africa, production has increased because they have increased the land use, the cropland area. So that's a big difference. And if we go on, if you have remember the amount of CO2 that are emitted by a hectare of CO2 by hectare of forest which is deforested, so we will see what happened. So here you have what happened. No, what? Okay, something happened. Okay, and can you hear me too? Yes. So here is what happened. For instance, this is a projection for what will happen. It's okay. You
Yes,
hear
sir,
me?
we can hear you.
Yeah.
We would request you to finish your talk if
Okay.
possible.
Well, I will try to maybe I will go a little bit quicker in some extent. But here is, for instance, the change in wheat production. But it's going to get reduced in most of African countries, except if you improve the seeds and if you improve the input. That's IFPRI study, and we show that you can maintain, and even sometimes with green points we can even increase sometimes the yields for in 2050 according to models, of course, climate change models. But that means that's a big difference between between the two diagrams. And that would be very important for decision maker to decide what to know what to do in, for instance, in Africa. But this type of message is not conveyed to them. So that's very important. You can also increase yields per hectare with different with irrigation and also with fertilizer. That's in Benin. So I'm trying to go fast. This is what we have said since more than 20 years. Well, to reduce, of course, we should not exaggerate. We're using a nitrate fertilizer because in some case you need to limit leakage for nitrate leakage to preserve the quality of water. And what came out recently is that you have to assess the situation of agricultural soil, not in spring as was usually done in temperate countries, but at the end at the end of a year, beginning of winter. So you will see how much nitrogen you have in the soil, and so you have to change and to assess the amount of nitrogen which are in soils much earlier than we thought before in the past, when you are concerned by so well, I say.
Excuse me, Professor Lideker, sorry to interrupt you in between. Sir, we are running out of time. Can you please conclude as soon as possible?
I couldn't understand you.
Sir, I am saying that we are running out of time. Can we please conclude with the presentation?
Let's say I'm trying to get quicker. So I already say that what the EU is doing in that.
Sir, yes, sir. Please continue.
Well, I'm trying to be quicker.
Sir, continue. Yes, sir.
So what I'm saying is what EU decided recently is to have more organic farming in Europe. That would decrease total yield by 8%, 8 to 20%. That's completely unresponsible, because it's similar to reduce for OPEC country to decide to reduce the production of oil. That means this would be increase the price and would be a particularly inappropriate recommendation in the context of Russian-Ukrainian war. So, for instance, this is you may see what these are. Egypt is a major importing country, when China and Algeria are importing countries, and Russia and Ukraine have disappeared from this picture. So, and they are afraid of having riots in the future in Egypt. So, like in the past. So it's very important. So I'm not insisting on that. These are main producer of nitrogen fertilizer, and what disappeared due to the recent war. And this is what we increase of production, but we have to satisfy to meet the demand of up to 2050. That means you have to increase the biomass production. So I'm not insisting on that. It depends on the country, that means at the end, the problem of UNFCCC inventories is that they don't take into account the avoided emissions So that's really a problem, because decision maker cannot decide what to do when if you look just at the emissions in the national inventory reports. So this is so. For instance, here is don't know if you have ever looked at the UNFCCC greenhouse gas inventories of your country. This is for France, but all these reports are available on the UNFCCC international international. So, but here what you have, I have removed the other. I just kept CO2, methane, and N2O, and of course, if you have this, you have a really total emissions are high. But if you take into account the biomass production, which is not in the reports, picture will change, because biomass, you have zero emission, but you need land, and you have here. We have poplar, we have a forest, average forest, you have poplar, and here you need much less land if you have some input, and that's very important. But this is a kind of information which don't exist, and if instead of having very high emissions here in agriculture, you have negative emissions if you take into account the biomass production. That means we should add this, and methane and N2O are two major greenhouse gases in agriculture. One from enteric fermentation, animal waste, and rice crops, and the other from mineral and organic crops. So fertilizer. So I'm going quickly, but I'm not going to insist on that, because it's how you different ways where you can reduce greenhouse gas emission. But one thing which is very important: methane emissions in this accounting system which is used today is 3 times less than what would be necessary if you consider that we have to stabilize the climate by 2050. But this cannot be changed, because it's a policy decision. This cannot be changed under the UNFCCC convention and negotiation and international negotiation. And that it's really a big bias so I'm going to skip that. So that means the difference between rice crops and animals is rice are photosynthesizing, and they can compensate to some extent the greenhouse gas emission during their production, but animals cannot compensate because they have no photosynthesis. So that's very important, a big difference that you have to keep in mind. You can increase possibility to well, I'm specific. So one important message also is to eat less animal proteins. But this kind of graph doesn't exist in IPCC summary for policymakers, and it shows what in wealthy countries you could divide animal protein consumption by 2 and use more vegetable protein. I'm not insisting on that because we have no time. So this is what we can bet in your country, but on land which can be spared, you can produce more biomass, and you can use it for to replace fossil fuel. And also, you should have a better management of forests. This is something like you in California and Australia, they have a lot of forest fires, but with different management, these fires would be less important, and you would be able to have more biomass. So news journals, I'm not insisting on that. So, but what do NGOs say? They give a message which are completely contradictory to that. They say, for instance, in Doha, they said in 2010 that developed countries should reduce the quantity of animal meat consumption. So that's correct. But they also say that we should get rid of mineral fertilizer, which is completely irresponsible when you look at what when you remember what I've shown, and that means to reduce to come back to yields like in before Second World War II, which is much lower than today. That means even with organic farming, the yield would be half as high. But in addition to that, there would be not enough organic fertilizer to do that. So this is a region you may know that it's an Indian journal, and they insist with rightly that European countries should not buy natural gas, liquefied natural gas at much higher prices, and to divert it from developing countries. So that's a good, but they never have any reference to fertilizer manufacturing, which of course nitrogen is used, and they say that in the future we may have organic farming when we no longer have no longer have methane, so natural gas. And so they say a lot of interest organic farming, they don't take into account the land which is necessary to produce. But we have but one world, and there is no additional surface. And just to finish, 50 years ago, Denny Meadows issued this book, Limits of Growth, and it was 50 years ago. And the same problem in his book is, as what I said, there is no mention of difference between countries. It's just global. So that means somebody in developing countries, in a poor developed countries, and a rich country, Europe or US, they are about to do the same things, and this is not clearly this difference is not clearly shown. So I'm not insisting on that. So if you want to have more, this also you can find more in this paper. And well, to finish, I said, would it be important to have a supplementary report for IPCC? Would it be useful or not? And what kind of suggestion can you make to improve that type of presentation, which of course was a little bit short? And thank you for your comments. And here is my email. If you have any suggestion and comments, positive or negative, please do send it, because it's an important issue for everybody in the north and the south, and perhaps it will help us to improve the situation. Thank you very much. I'm sorry to have been too.
Thank you very much, Professor Decker. We are obliged to have you online. Due to some reasons, Sir was unable to join us physically, I guess due to health issues, but Sir has joined us online. Thank you very much, Sir. Thank you very much, Professor Decker.
Okay, I would like to ask one question.
Sir, we have a question for you.
Yeah, sure.
What would be the effect of the greenhouse gases of the Ukraine war on the greenhouse gases? Is it possible to have some idea that what would be the effect of Ukraine war on the greenhouse gases?
Sir, did you got the question?
No, I did not. Can you repeat it, please? Can you repeat it? Because
yes, sir. Sir is
not clear for me.
Okay, you are able to hear me, sir?
Yeah, it's okay.
So I am asking you that what would be the effect of the Ukraine war on the greenhouse gas emission?
Well, it's disastrous because on one hand, all the policy for land use and land use change will be changed. The yields may decrease. In North Africa, in Algeria and in Egypt, they will have major problems if the war is going on. And even this morning, I heard that in the US, they are allowing and again to increase the oil drilling in the US because the price of oil has increased too much. And so they are exactly doing the opposite of what should be done for to mitigate climate change. So it's really a pity, and we only can wish that the war will end as soon as possible. This is for to allow farmers to have proper proper yields and to limit famines, and so that's really a huge problem.
Okay, sir. Thanks a lot.
Thank you very much, sir. We can disconnect you now. Thank you very much, sir.
Thank
A
you.
huge
You're
round of
welcome.
applause for Professor Decker, please. Thank you,
Thank
sir.
you. Thank you. Don't forget to send me email
Sure, sir. Sure. I'll share you the email. Thank
and
you, sir.
comments to improve. Of course, you have not been able to listen
Right.
to everything, but you get a flavor, and so you should tell me if it's necessary to go on with this objective or not.
Sure, sir. I'll share the details. I'll share the things. Yes, sir. Thank you, sir. Goodbye, sir.
Okay. Bye bye.
Hello everyone. Myself Pooja Verma, third year student, Department of Biotechnology from Anandini College. I am grateful to you all for giving me this opportunity to present my research paper, which is all about ethnomedical flora for treatment of asthma treatment in district Agra, Western Uttar Pradesh. Asthma is not a quiet kind of condition. Symptoms can arise anywhere and everywhere at any time. Did you know those who have it are stuck for it life? Doctors are not sure why some people are more prone to getting it than others, but they believe it is a mix of genes and environment factors. Now, let's see what is asthma. A condition in which person's air may become inflamed, narrow and swelled and produce extra mucus which make it difficult to breathe. It is a chronic inflammatory. By the end of 2005, 25 genes has been associated with asthma. In 2006, more than 100 genes are associated with asthma in one scientific association. Signs and symptoms are more important. How can we cure it? Shortness of breath, cough, chest pain, wheezing sound when you are breathe, walking at night, waking at night due to asthma symptoms. Asthma develops are infection like cold and flus, allergies such as pollen, dust mite, animal furs, smokes, fumes, and pollutions. Medicines, particularly anti-inflammatory painkiller like ibuprofen and ice cream. Smoking during the pregnancy after delivery is associated with a greater risk of asthma-like symptoms. Low quality of air from environmental factors such as traffic pollution or high ozone level can associate with asthma development. According to the Center of Disease Control and Prevention, it affects 1 in 13 American, includes 20 million adults ages 18 and over 5.1 million children under the age of 18. In 2019 survey, they concluded 46,500 people are died due to asthma in India only. Now I am going to share with you all one of the methods that can be used to cure ethnomedical. So basically,
2 minutes.
It is a study of ethnomedicine. It is a study of comparison of traditional medicine based on the bioactive compound in plants and animals to practiced by various ethnic groups and especially those with little access of Western medicine. Example, endogenous. जैसा हम सब जानते हैं कि हमारे भारतवर्ष प्राचीन काल से ही आयुर्वेदा, जड़ी बूटियों और भूमि रहा है। हमारे वेद पुराण इस बात की पुष्टि करते हैं। रामायण में हनुमान जी द्वारा संजीवनी बूटी लागे हुए पर हमारे जैन भाई जो मेट्रोजाइज होते होते अपनी आयुर्वेदिक परंपरा से कहीं दूर हो गए हैं। अपनी प्रेजेंटेशन के माध्यम से मैं एक छोटा सा प्रयास करूंगी अपनी परंपराओं को पुनर्जीवित करने का। उसके कुछ फोना के बढ़िया एग्जांपल मैं आपको बताती हूँ। कैथा जो कि हमारे लोकेलिटी टूंडला आगरा में मिलता है। इसके यूजर्स है लोकल प्रैक्टिशनर। इसको हम यूज करते हैं लीफ मिक्स्ड विद हनी 10 ml by black peppers given orally twice a day in customer bronchitis.
Please conclude.
To bring us to the end of the presentation, I would like to summarize by saying I.S.G.B.R.D is including in the WHO Global Action Plan for the Prevention and Control of NST and the United Nations 2030 Agenda for the Sustainable Development. Now I would interest some questions from you all.
Any question? Query? Okay, thank you. Next 5 minutes only.
Good afternoon to all. My name is I.S.G.B.R.D. I am presenting correlation and path analysis of yield and qualitative traits in rice. Correlation was performed to explore the degree of relationship between the characters, and it will help to understand how improvement in one character will simultaneously changes in the other character. And in path analysis splits the correlation coefficient into the measures of direct and indirect effect of a set of independent variable on the dependent variable. And this experiment consisted 67 rice genotypes obtained from the rice improvement program from the Madhya Pradesh. And here are some observations recorded. These are the 20 quantitative and 14 qualitative traits. And you can see in the left slide, you can see the anthocyanin pigment coloration of the leaf and basal leaf sheet coloration in the right. And here you can see the difference in lemma and peda color. And here some diverse rice genotypes. These are the diverse variety which are having different length of cones. I performed 3 statistical analysis: correlation analysis, path analysis, and genetic divergence. And I am concluding here my results. As correlation coefficient, total 7 genotypes, 7 traits are showing negative correlation, as well as 8 traits were showing positive correlation, where I marked as a red, showing the positive significant correlation towards the yield. And selection from these traits can be further using in the further yield improvement program. And
2 minutes.
Path coefficient analysis show the number of spikes per panicle. And here the diagonal red color showing the indirect effect and blue one showing the direct effect of the grade. And this is the t-square statistics, which I have concluded these genotypes into 12 clusters. And I am concluding the slide that all traits selection can be effective on the basis of significant positive correlation with number of productive tillers and so on. And path analysis revealed that the spikelet fertility, cochlear length, biological yield per plant, panicle yield per plant, and the maximum positive direct effect on grain yield per plant. Hence, for the enhancement of yield, these traits should be given due to emphasis in breeding program.
ये जो है आपका ये जो ये आपके phenotypic traits हैं उसी से आपकी genotypic analysis है।
Thank you very much. Now I will request the chairperson of this technical session and the co-chairperson to please associate with him and conclude the session.
Myself, Subhash Chandra, Senior Scientist Horticulture, KVK Projawat, CSA University, Kanpur. First of all, here research paper delivered by the Sweta Kuswa, I.S.G.B.R.D, and he has totally explained about the causal organism of HIV and correlation with the HIV and tuberculosis. Second presentation by Mamta Sahani and paper totally related to the biotechnology, research related to the genes structure and the chromosomes. All the research scholars, scientists and professors have listened with the consultation of the mind, constant of the brain also, and he has emphasis on the monocots and dicots condition of the seed. Third paper presentation delivered by Dr. Satish Pari, PhD scholar from I.S.G.B.R.D, Hyderabad, a line and tester analysis for grain yield and contributory traits in palm millet. And I would like to say that this was a very excellent presentation from the, he came from Hyderabad, I.S.G.B.R.D, and presently he is working in the Rallis India Limited. Now, I would like to say that Mr. Rahul Singh, MTech Biotechnology topic is silico analysis of non-tuberculosis Santium by granuloma proteins related delivered. It was also very effective and knowledgeful lecture. Fourth lecture delivered by the paper presented by the Dr. I.S.G.B.R.D, ICR, Bioscience, and he has told genomic assisted breeding for chickpea improvement and very excellent research paper delivered by them and especially focused on the line presentation. Mr. Tulsidas Viswas RBS Technical Campus delivered the research paper here that was totally related to the food processing and fermented production quality wise. Mr. Keshav Dev Singh RBS Campus with Pune Agra also presented the paper that is related to the medicinal and spices, antimicrobial and antimicrobacterial position of the topic. Deepika Bharde has also delivered here research paper that is totally related to the correlation and path analysis of yield and qualitative traits in rice. She has also presented in the good position and all the scientists, research scholars and professors have listened on the behalf of the regulatory sensitivity punctuality. Pooja Verma, Anand Singh College recently taught the lecture or research paper, that is also very good. The topic that is known as side effect in, Dr. I.S.G.B.R.D, MP Dhar district, MP. He has delivered the lecture very well with the effective methods of the research and teaching. And all the research scholars and scientists or professors have taken the maximum interest because it was related to the human body, especially fluoride content and fluorisis. So, side effect in fluoride, water and prevention control of fluorisis. Now, before these two lectures, here Dr. Arthur Ridecker, NRAF, I.S.G.B.R.D, Asia and Africa, delivered the lecture related to the carbon theory and related to the increasing of the production in South Africa and Asia also. He has discussed about the day by day going to change the climate factors in all over the world, and also guided the research scholar and professors or scientists here by them that how can we increase the production of the wheat in the different negative circumstances of the change of the climate of global warming. All the audience who have listened here carefully, I would like to thank, heartily thanks to all of you. Thank you.
Now I would request Professor Rawat and Professor Chonka to please conclude with the poster presentation session.
Good afternoon. Good afternoon to everyone. So we had a poster session outside this auditorium, and total 4 number of posters have been presented by research scholars, and all the posters are well designed and very actually they have done hard work to present their work. First was by the Ms. Deepika Patre from, I think she is from Jabalpur University. She has presented work on genetic diversity analysis in aromatic and non-aromatic germ class of rice, which is one of the very good work for education of good traits, and phenotypic classification she has done it in rice. And second poster was by Dr. Ram Krishna from CMAP Center, Institute of Medicine Aromatic by Lucknow. And the title of his presentation was Identification and Cloning of Terpene Synthesis Gene from Vetiver Transcriptome. That is a very good piece of work. Just to you can see how a gene can be identified in the pathway, and that gene can be incorporated in one of the vetiver for the high yielding essential oil content. And Dr. Prasad Yadav has presented work on development of induced herbicide resistance in the Indian mustard to control by the wheat Orobantia. He has designed a very good experiment on that, and the last one was presented by Mamta Sani of the Institute to study the effect of foliar spray of thiourea on growth and yield quality of wheat. So all the 4 presenters were well answered the question also, and they have a deep information and knowledge about their work. And in the last, we enjoyed the presentation. Thank you very much.
Thank you very much, sir. We conclude with this technical session. Tomorrow, the valedictory session will start sharp at 11 in the morning. All are requested to please assemble in the Dr. APJ Abdul Kalam Auditorium Hall of Hindustan College of Science and Technology, Khared. All the awards and certificates will be distributed there only, participation certificates and award certificates with the mementos. Apart from this, the important information was regarding the trip which is planned tomorrow for Agra. You need to inform about your trip to our volunteers, Ayush and Anshika, regarding the same. Thank you. We have proposed the high tea plan, so all of you may please gather for the high tea and then move towards the hall for the cultural event. Thank you.